Thank you very much, and welcome everybody to the annual conference of EAMDA Medical Day. I would like to start with some housekeeping. When you don't present or speak, please keep your microphones on mute. The questions and answers will follow each of the presentations. And finally, I would like to ask you all to agree to record this event. Is anybody against this? If not, thank you very much. So I'm Dorica Dan, and I'm going to be the moderator of the Medical Day of EAMDA Conference 2022. I am the president of Romania National Alliance for Rare Disease and a board member of EURORDIS since 2007. And currently, I'm one of the vice presidents of EURORDIS. So EAMDA is the European Alliance of Neuromuscular Disorders Associations and works to support and improve the quality of life for people affected by neuromuscular disorders. This conference is an opportunity to exchange knowledge and expertise and experience, and is funded by EURORDIS, Novartis, and Roche. For the official opening of this day, I'm honored to invite to speak Arabella Caline, the president of EAMDA. Arabella, the floor is yours. Thank you.
Thank you so much, Dorica, for passing me the floor. So welcome everyone. I'm very excited, but I must admit also very nervous because, you know, it's the first time I kind of organize such a big event as president of EAMDA. So I want to thank all of you for participating. You are very important partners, member organizations, medical experts, collaborators, and stakeholders. So we very much thank you for your participation. With your permission, I will share my screen because I want to have a short presentation of EAMDA. So, okay, can you see my slides? Okay, so if everything is running okay, again, welcome to our International Annual NMD Conference 2022. I'm, as mentioned, Arabella Caline, president of EAMDA, president of Neuromove CMT Association in Romania, which is an association, a national association dedicated to patients with Charcot-Marie-Tooth and other neuromuscular conditions. I'm a board member of the European CMT Federation and their Eastern Europe ambassador. I'm also a EURORDIS summer and winter school graduate and a UPATI fellow myself. I'm a philology graduate. This is my basic training, but then I specialized in several fields like training and career and vocational counseling, especially for people with disabilities. But first of all, I'm also a Charcot-Marie-Tooth patient and mother of 2 sons with the same disease. Let me just check if my slides are moving. Okay, so a little bit about EAMDA. EAMDA was founded in 1974 by organizations from 9 different European countries: Belgium, Denmark, Germany, France, Great Britain, Italy, Netherlands, Sweden, and back then Yugoslavia. EAMDA was first established in England, then later on it was moved to Malta, and now EAMDA is established in Ljubljana in Slovenia. So our office is currently in Ljubljana, Slovenia. At that time, when EAMDA was established, a constitution was accepted with the following major aims: to develop help for all people who suffer from muscular dystrophy and other related disorders, to improve their physical and mental conditions, both with regard to their social and professional possibilities, to support research into the causes, treatment, and cure of this group of diseases. So this was the major aims that EAMDA proposed even from the beginning. Nowadays, we are 7 executive committee members that are running EAMDA. So I do have two vice presidents, one from Czech Republic and one from Bulgaria, Dona Jandova and Jana Popova, that are very dear colleagues and very passionate patient advocates themselves, living with an NMD themselves. Actually, most of us are. That's why we are here running EAMDA. Tomislav Gol is our Secretary General. He's from Croatia. He's also a patient with CMT. Ivanka Jovanovic is from Serbia. She's an executive board member. Mojca Susek is our lawyer in EAMDA and our partner from Slovenia, our executive board member there. And Katarina is from Croatia, again, a young but very dedicated executive committee member. So all in all, I'm very happy to have this wonderful team besides me. And one person that I actually just realized I didn't include it in my presentation, but I have to tell you about her because she's basically helping me on a daily basis. İpek from Turkey, who is here with us on Zoom. She's our secretary and our communication manager. EAMDA is trying lately to change a little bit the communication with the different stakeholders, and we are trying to improve in this field. We just expand our social media channels of communication. So İpek is to blame for all this very good work. Thank you, İpek. And EAMDA also focused a lot on the network of medical experts that, you know, we cannot live without interacting with doctors, and sometimes we have to rely on medical experts so much, especially as patients. So in 1999, EAMDA, together with AFME Teleton, laid the foundation of the European Neuromuscular Center, ENMC. I'm sure you already know about ENMC, which later on, of course, became independent body. But we are very proud that EAMDA had a contribution in forming in ENMC. EAMDA annually organizes these large international conferences dedicated to neuromuscular community and to medical experts in the field. And it is usually a conference that we choose to structure on 2 days, a medical day and a social day, because we want to also have the opportunity of connecting medical experts and stakeholders in the field and putting the basis of this active European network in the field of NMD. And we also want to focus on the life of everyday life, actually, of patients themselves. That's why we choose, for example, this year to get in front of you a large variety of organizations that have a very good practice in the field of helping patients with NMD. So yes, these are some pictures. Of course, we had the privilege of organizing this international conference in person until COVID. Everything changed with COVID, but hopefully we will be able to maybe do it again live next year. So now we are still sticking to online format. We do have EAMDA had in the past, before COVID again, a specific focus on social aspects, and we didn't want to leave behind the young generation. That's why EAMDA used to organize regular youth camps, which were basically summer camps for young people with neuromuscular conditions. And in this camp, we mainly focused on rehabilitation activities, social activities, but also educational programs. Because even if these patients are young age, they need to be educated and helped in better managing their condition. We also had a very interesting program last year, which was a 6 months program. It was our basic desire to raise awareness about NMD in Europe, and especially in eastern part of Europe, where, you know, unfortunately, there is this discrepancy sometimes, not always, but sometimes we can still feel this discrepancy between Western and Eastern Europe. And that's why we are also doing a lot of work in trying to raise the awareness and the expertise in this part of Europe. So we organized this educational radio program, which was running from July to December 2021, and it was actually it was in 6 countries, but also put online so everyone can have access to it. So we coordinated it in Romania, Croatia, Slovenia, Poland, Czech Republic, and Bulgaria. We partnered with Radio 92 FM Croatia. We basically prepared a wide educational program that was broadcasted through Radio 92 FM Croatia, but then also put online so the general public can access all these educational materials and all these lectures that we had. We disseminated it online in national languages, but also translated it into English. We had more than 40 specialists involved, which was amazing because we could in this way connect a lot of experts. We organized this year in June 2022 our first think tank meeting. It was not only because I was quite new as I had become president of EAMDA, but also because we really wanted to reorganize a little bit EAMDA structure in the sense that we wanted to aim more on those activities and projects that will respond to the specific needs of member organizations and our NMD community. So we organized this event together with member organizations, with partners, and also with farmer representatives. As you know, EAMDA has worked with farmer representatives because we have common goals. When it comes to new treatments, it's them that inform us and so on. So we had this online event. Then we also went to specific disease-specific projects. And we organized in August 2022, sorry. So again, this year, we organized during the SMA Awareness Month, a complex communication plan that we were running, which primary aim was to actively involve the participation of NMD patients in Europe. So we wanted to create something that was more specific to patients themselves, so they can get a feeling that they are involved in a very personal level. So we organized our NFT art gallery, which was is dedicated to SMA patients. It is actually a unique event in the field because from our understanding, an SMA art gallery wasn't ever organized elsewhere in the world. So we could say it was quite an innovative project even for us. We were basically promoting the SMA community, but the focus was on their artistic abilities. You could see here some pieces of art that we collected. But I will also have the pleasure after my presentation to let you take a look at 2 very short videos that will explain more our project. We were very happy that we had very positive feedback from all over Europe regarding this project. We also organized an SMA webinar, a European webinar with a very nice agenda. We were very happy to have also an MEP with us. And we were also very happy to have some very nice organizations and some very wonderful presentations, a very good practice model, especially, for example, in Poland. SMA Foundation Poland is doing a great work there. So we really had a very nice event that you can also visit it and see it online because we put it on our YouTube channel. We had then in September 2022, we had our AGA meeting in Ljubljana. It was actually the first time we did AGA in a hybrid version. still due to COVID, but not only. So we had some guests with us, some members that were online. And we also managed to get together in Ljubljana, which was again amazing and very productive because we managed to brainstorm a lot and find some new ideas and come with some very new projects that you will hear about them later on. So keep posted and stay close to EAMDA. But of course, in our belief, collaboration is the most essential thing. And actually, it's like something that EAMDA was very focused on, even from the beginning. We think that collaboration is essential in order to build a better future for the NMD community. That's why we are having regular meetings and we are trying to gather the member organizations and to get them very actively involved in EAMDA. This is our plan for next year, for example. Thank you so much for your attention. These are some of the ideas I wanted to present you about EAMDA. And you can always contact us on that phone number that you see on the screen or on the secretariat email. You can easily reach us. And now in the end, I also want to personally thank our sponsors. And later today, you will see a short video with a very, very short presentation of the sponsor. So we are very happy and honored that we had sponsorship for this conference from Eurordis, from Novartis, and from Roche. We couldn't have coordinated all these international conference without their support and help, of course. And regarding coming back to our NFT art gallery, I would ask our IT colleague to play. We prepared two special videos for you that they are very short, but for you to see something that it's very close to our heart, because EAMDA has also been, split it in a way, trying to balance also the personal side of engaging with patients and of course medical part and research. So especially for the doctors that are here present, you will see a different part of a different side of your patients. So thank you very much for your attention. Steffi, if you can play the videos, please. Thank you so much.
There goes another perfect moment where I should have said what's on my mind. A few words would maybe make a difference, and then maybe I could call you mine. 'Cause I've been holding it in for forever, and I can't hold it in no more. It's time to spit out the words right onto the floor. Maybe you've had your suspicions. I've tried to play it so aloof. It's safe to hide behind the lie, but I think it's time you know the truth. 'Cause I've been holding it in for forever, and I can't hold it in no more. It's time to spit out the words right onto the floor. I see me in a different light. 'Cause I've been holding it in for forever, and I can't hold it in no more. You gotta know that I love you, baby. That's what this song.
My name is Polina Kozenkova. I'm 11 years old and I'm from Ukraine, Kharkiv. My hobby is drawing. My artwork named Sma and Me. With this work, I want to show you that Sma not only touch my body but also changed my character.
My name is Nina Santiago. I'm from the US. I'm 37 years old and I have SMA type 2. My piece is called Breathe, and it is about embracing the beautiful chaos. As long as you're still breathing, you can see it, and that's beautiful, and that's a blessing.
Hi everyone, this is Sedat Karakaya. I'm from Turkey. I'm 25 years old and I have SMA type 2. It is just reflecting the idea of the SMA. I wanted to draw myself and my favorite TV series. I hope you liked it.
Hello, my name is Marcel Phillips. I have SMA type 1. I'm 16 years old and I am from Slovenia. I love life and I enjoy it immensely. Join us. Help us build a better future for people living with NMD.
Very touching video. Thank you so much. And it's very important for everybody to see real patients and to find out more about the real life of the patients. For our next presentation, our next speaker is Eglis Gonzalez, and we'll speak about Safe Polymed project. It is a European research project with the main goal to increase drug treatment safety, to provide physicians and pharmacists with innovative tools to define, assess, and manage drug interactions. Overall, the tool will not only empower healthcare providers, but will also educate patients and citizens on how to safely manage their drug treatments. Eglis Gonzalez, the floor is yours.
Good morning. I am Eglis Gonzalez from Leukanet and Padvocate, and I'm going to present to you today a new project in which we are being involved. It's the Safe Polymed, improving safety in polymedication by managing drug-drug gene interactions. This is a project part of the Horizon funding program. It's a long time project of 42 months, and it started just June this year. It's a very international project within Europe. Our coordinators are located in Germany from Saarland University. We have from many other countries clinical partners that will be supporting the project. As patient organization is us, Leukanet, led by Jan Geisler. We have a group of experts in artificial intelligence and machine learning, and also in health information system, working for some of the work packages. And our great group of project management that helps us a lot in the coordination part. So this project raises because of the current situation in polymedication. And there is a very high number, about 200,000 deaths related to adverse drug reactions. The most affected people for these type of reactions are the elderly population, because they usually take more than 5 drugs at the same time. And of course, on an economic point of view, very big amount of money is spent in healthcare related to response or hospitalizations or management of patients that come because of these adverse drug reactions. So we have more than 60 and 65% of these reactions that are actually related to drug-gene interactions and drug-drug interactions. So these two components are the basis of this project. Drug-drug interactions is not the only issue, and drug-drug interaction has been a little bit more studied and more considered in the last years. But drug-gene interactions have gained some inputs and information, and a lot has been known in the last years. And one of the problems is, for example, here's an example in a way when one drug is given with another one or with two other ones, the metabolization of drug number one is very, very affected. And this can transform into a very complex reaction in the patient, also leading then to death of patients. So this is the main core for this project. That's why we call it then drug-drug gene interaction in a complete way. So with this project, we expect that there is a high number of drug reactions, adverse drug reactions that are preventable, and we expect to reduce that number. And also, because of this reaction, there is a poor adherence of patients into their therapies, which is understandable. And we would expect then to increase adherence when we kind of help control this type of reactions when people are getting more than one drug. And of course, any economic improvement that we can make will be also helpful for our healthcare system in Europe. So an overview of the project, we will be having from real-world data a prediction of risk for each patient. We will be developing a patient-reported outcome based on safety and not disease-related. We will have a dose precision model, and all this will be loaded into a medication management center that will be available for patients and clinicians. By the end of the project, we will be performing a clinical case study in which all these tools will be then validated. So as main objectives of this complete project, we would like to increase the patient safety and equip healthcare providers with innovative tools that help them efficiently manage polymedicated patients. We want to increase the citizen participation and resilience in healthcare, empower citizens to proactively manage their own healthcare and be aware of their own conditions and situation, to support the development and uptake of innovative healthcare system in Europe, to increase the patient safety towards a personalized treatment plan. So in a more detailed way, I will explain. So we will be generating an evidence-based risk score model, which is part of work package 1. We will be using 3 biobanks that are available for us. And then using machine learning, we will be generating this score model that can help us identify these high risk or low risk individuals. Then for the patient reported outcome is part of work package 2, but work package 2 is not just about the PRO development. we actually empowered patients, generating some trainings that are actually not only directed to the patients, but to the clinicians too. So developing then the PRO and increasing the participative actions to improve then the quality and safety of the patient management. So these tools generated here, PRO, the trainings, and will be loaded in the Medication Management Center. And to do this, we need the generation of a patient engagement hub. That is what it is. It's just a group of patient organizations that have the experience, the knowledge, the capacity to provide the patient point of view, to express the patient needs, and whether all these tools that we will generate and do adapt to the citizens and to the patients that really need these tools. So this patient engagement hub or these patient organizations will be helping us contributing to the development of the training courses, the PRO, and also a constant feedback on the Medication Management Center, making sure that everything is patient-centered and that is understandable and that it's actually covering the needs of the patients in these topics. So then we have the precision dosing model that will be then performed by Work Package 4. And in these cases, by modeling, we will be identifying any kind of dose adjustment that may be necessary considering the patient's own genes and patient's own condition and any, of course, drug-drug interaction that can be identified. So this will give us also a model of system kind of an alarm whether there is a risk related to this specific patient. So everything that I just mentioned will be then loaded in the management center, and that is Work Package 3. So we have the risk score or the risk model, we have the dosing model, and we have then the PROs that will be available in this management center. And it will be managed by patients and clinicians in order to increase the communication between these two. So then we have our clinical case study, which is called EMPA-SAFE. It's empowering patients to improve safety in polymedication, and that is part of Work Package 5. We will be having 4 locations, collecting then patients, and all the tools that are going to be developed during this project are going to be validated in this small study. So we will be seeing whether we have the ability to identify any drug-drug gene interaction and whether that has any effect in the adverse drug reactions. And of course, a calculation of an economic consequence. So in a more general way, we have all these 5 work packages, like I said. So the risk score, PRO, and those model will be loaded in the management center, and all of these will be then validated in the clinical case. Then above us, we have our project management group, which coordinates the whole project and makes sure that everything is done adequately and in time, which is very important. And then we have also for the communication and dissemination, Work Package 7, a group of people that are in charge of generating all these materials and showing that we have visibility and increasing then the impact of the project within the population. So In general, the key results from the project will be in two phases, like supporting the citizens to manage their own healthcare. So we will have the training courses. That is actually a tool that is meant to be used even when the project has ended. The Medication Management Center also in a sustainability program, the idea is that it can be used further. And the Patient Engagement Hub is our very big base of experience and information, helping that this project is really directed in a patient-centered way. And on another level, also trying to give some tools to the healthcare professionals that can help them manage more efficiently polymedicated patients. So we will have this risk score model, the PRO, and the dose precision model. So that is more or less the results of expected results of this project in a nutshell. Currently, the project basically just started, like I said, in June. We already had our first face-to-face meeting by the end of November. And it was a very fruitful meeting where we got the opportunity to meet and learn from all of the professionals that are actually contributing to this project. And well, there is still a long way to go with this. And I would like to invite you to visit our webpage, where you can find probably a little bit more extended information than what I just gave you. And thank you very much for your attention. I'm sorry I couldn't be there live, but if you have any question, any comment, any suggestion, I'm happy then to answer any of them, and to just contact me for that. Thank you very much.
So I will ask my IT colleague to wait.
Did you know that Charcot-Marie tooth is the most common rare disease in the world? CMT is a hereditary disease that affects the peripheral nerves controlling the muscles. It is not a muscular dystrophy that directly affects the muscles. There are different types of CMT, CMT1A, CMT1B, CMT2A, etc., and the development varies with the form of the disease. It concerns more than 2.8 million people worldwide, one person in 2,500, men and women from all ethnic groups and origins. The disease has been identified 135 years ago by Jean-Martin Charcot, Pierre Marie, and Howard Henry Tooth, and it is still a rare disease with no cure yet. There are a few signs that characterize CMT and that vary from a person to another. Loss of sensation in feet and legs, leading to falls, difficulty to walk, aching feet, difficulty to lift feet or ankle, frequent tripping and problems of balance, weakness in hands and difficulty to grip things, presence of hammer toes, awkward steps higher than normal, weak legs and muscles, loss of the ability to run, problem with sleep apnea and breathing troubles. On average, the first symptoms are felt around 21 years old, but it can be identified earlier, during childhood. Sometimes, due to a lack of awareness of the disease, the correct diagnosis comes late and can take, on average, up to 10 years. Still, after this first diagnosis, only 42% of the patients have received all the information about the risks in weakness in muscles, feet deformity, sensory disorders, and possible sprains. With CMT, daily life is significantly changed, even if it is a slowly progressive disease. Often, patients' feet become deformed, and there are several types of foot deformities: hollow foot, valgus, equinus, steppage, and on and on. They wear insoles or orthopedic shoes, and sometimes require a wheelchair. To reduce the effects of the disease, health professionals must treat the ills, try to understand the causes. Many of the neurologists, orthopedic surgeons, or podiatrists that are aware of the CMT disease agree on the importance of a multidisciplinary team involved in the care process of the CMT patients. General practitioners, neurologists, surgeons, podiatrists, physiotherapists, psychologists, and so on and so forth. It is also very important to work with the patients and encourage them to share their everyday life, to better understand their disorder and be able to guide them and propose adapted therapeutic solutions. Since 2017, the European CMT Federation organizes the Awareness Month in October. The ECMTF is a nonprofit organization that works also on the international level and is the main interlocutor of the CMT European institutions and national associations. Find out more on ecmtf.org. Charcot-Marie tooth disease remains unknown. Awareness must continue for a better diagnosis and care of the patients. You can help us by sharing this campaign. Tell us your story on social media with the hashtag CMT Awareness. To know more, check the website of the campaign, cmt-awareness.com.
So coming back, just wanted to tell you a little bit about the video we just presented, because as we can all see, most neuromuscular disorders have some common issues. Most patients with neuromuscular disorders eventually face the same challenges at one level. So we really wanted to also include in our program this video about CMT. It was actually a video that was made up by the European CMT Federation in the attempt to raise awareness about this condition, which is very common, by the way. It affects 1 in 2,500 people. In my family, we are already 3 generations, 14 people from one family only. And it can be very invalidating. You can have mild walking problems, or you can get into a wheelchair at a very young age. It has many forms. It has more than 100 genetic mutations. And I'm very happy that we have a lot of doctors with us today, because in this way, they can learn a little bit more about CMT. So thank you for the opportunity of sharing this video also. Now I will pass back on to Dorica to present our next guest. I know we are a little bit before the schedule, in front of the schedule, but I really hope that our next guest is going to be able to present a little bit sooner. So Dorica.
So thank you very much. Indeed, as many other rare diseases, this disease is proving that late diagnosis can affect the life of the patient and the life of the family. Our next topic is Duchenne muscular dystrophy, which is a severe type of muscular dystrophy that primarily affects boys. Our next speaker, Dr. Ivan Lehman from Croatia, will speak about Duchenne muscular dystrophy. He works as a pediatrician at University Hospital Center in Zagreb, Department of Pediatrics, Institute of Neuropediatrics, and meet in his daily activity children with neuromuscular disease every day. He published many scientific papers, scientific articles dedicated to neuromuscular diseases. More about this topic from Dr. Ivan Lehman. Welcome, Dr. Ivan Lehman, and thank you very much for sharing your experience with us today.
Yes, thank you. Thank you very much, and good day to everybody. It's really an honor for me to be participant in this conference. I will give today an overview of Duchenne muscular dystrophy that is really frequent found among muscular dystrophies. And also there is some improvement in the therapy of Duchenne muscular dystrophy that has been seen in the last few years. When we talk about muscular dystrophies in general, it is a group of inherited usually non-inflammatory disorders that affect muscles, and they are really rather different. They can present from early childhood to adulthood. Distribution of muscles that are affected can be different in certain types of muscular dystrophies. Also, the mode of inheritance can be different. Lots of muscular dystrophies can affect heart muscle. That is also another medical issue that can be a really important topic when discussing muscular dystrophies. Among all muscular dystrophies, Duchenne muscular dystrophy is actually the most common one, and its milder variant, Becker muscular dystrophy, is the second most common muscular dystrophy of all. When we talk about Duchenne muscular dystrophy, it is really a progressive disease, a severe one. And as you can see on this graph, actually each patient deteriorates, and it happens really quickly. The first symptoms manifest usually, clinical symptoms manifest usually around the second or the third year of life, usually problems with the gait, with climbing the stairs, frequent falls, and it is progressive, and patients become wheelchair bounded at the age of about 12. Respiratory insufficiency is the main problem, and it happens usually in the late teens or early 20s. When we talk about the cause of the disease is caused by the mutation in the biggest gene in human genome. It is called the DMD gene, and it has 79 exons. And as you will see a little bit lately, it is actually a problem for some sort of the therapy. The gene is placed on X chromosome, and the disease is inherited X-linked recessively. It means that women are carriers and the boys are affected. The protein is called dystrophin, and its function is to connect contractile filaments to cell membrane complex. And without the dystrophin, the cell cannot keep its integrity. The frequency of dystrophinopathies, and when I say dystrophinopathies, I mean a spectrum of diseases that is caused with malfunction of the dystrophin, is 1 in 6,000 to 1 in 5,000 newborn boys, and more than half of them have the most severe type, Duchenne muscular dystrophy. Prevalence of DMD is 63 per million, and it is much more frequent than its milder form, Becker muscular dystrophy. Women get DMD really rare, less than 1 in million, and usually it is due to some chromosomal rearrangements or other genetic causes that are rare. When we talk about women carriers, even some of them, up to 20%, can have some muscle problems or weakness, and up to 17% of women carriers can develop cardiomyopathy. The disease is inherited, but 1/3 of all mutations arise de novo. That means that not all mothers of boys with Duchenne are carriers. And what is the goal in the future? The goal is to get optimal care of patients with Duchenne muscular dystrophy. Optimal care means better survival in the end and better quality of life. How can we achieve that? It's by following standards of care, especially dealing with cardiopulmonary complications, but also, as I will show later, we can expect some improvement in pharmacotherapy of Duchenne muscular dystrophy. Now I will say a few words about mutations. It is important for the establishing diagnosis, but also for the treatment. So as you can see, the spectrum of the mutations is almost the same everywhere in the world. Some mutations are more frequent than the others, and the most frequent mutations are large deletions and duplications. Those are deletions that affect the whole exons of the gene, or duplications, but also there are some other mutations, small ones like small deletions, small insertion, and point mutations that are also important for the therapy. But among all the patients, majority of the mutations happen between the exons 45 and 55, and it is called the hotspot region for the mutation. So we have lots of mutations, thousands of them. So there is a question, is there a connection between genotype and phenotype? Can we predict the phenotype, a young boy diagnosed with mutation in dystrophin gene? Actually, in most cases, we can. And the key is if a mutation causes a frame shift in reading frame. So the mutations that cause frame shift usually cause the patient to have Duchenne muscular dystrophy, and mutations that are in frame usually cause Becker muscular dystrophy. But there are a few exceptions to that rule. So here is an example. This is normal dystrophin, and if a patient has deletion that is out of frame, then the synthesized protein will not be functional, and the patient will manifest Duchenne muscular dystrophy. And if the mutation is in frame, then the protein will be shorter and it will not function perfectly, but it will function at some level, and the phenotype will be a milder one, Becker muscular dystrophy. And I want to say here that nonsense mutation, that is a single nucleotide change that causes mutation that leads to premature stop codon, causes a severe type Duchenne muscular dystrophy. I will explain that a bit later. Can we check when we have results of gene testing? Can we check it if mutation is in frame or out of frame? Yes, there are some reading frame checkers on the internet that are freely available. And if you put in starting exon and the last one that is deleted or duplicated, then you can check if the mutation is in frame or out of frame. But I want to emphasize here that it is not 100% as you saw earlier. There are some patients that have, especially for certain exons, that can have out of frame deletion, but still milder type, but those are rather rare. And what happens with the muscle when there is no dystrophin? When there is no dystrophin, cells goes through certain pathophysiological processes that finally lead to a loss of muscle fibers that are replaced with fat and connective tissue, usually due to ischemia of the muscle, oxidative stress, influx of calcium also, and inflammatory response that all lead to cell death. And then the muscle fibers are replaced with other tissue that we can see on muscle biopsy. Also, on muscle biopsy, we can check with immunohistochemistry or other methods if there is dystrophin present at all, and so we can prove that the patient has dystrophinopathy or Duchenne muscular dystrophy if there is no dystrophin at all. Clinical presentation. First of all, laboratory signs and histological signs are present since birth. That is something that maybe in the future we can use for newborn screening for Duchenne muscular dystrophy. Nowadays, there is still no adequate therapy for all patients with DMD, and criteria are not completely fulfilled for newborn screening to be introduced everywhere. So the first signs are muscle weakness that present usually at the age of 2 or 3 years of age. It affects primarily proximal muscle on lower extremities. So boys have difficulties in running, jumping, climbing stairs. They are slower than their peers of the same age. The gait is typically waddling, and boys develop lumbar lordosis, and there is pseudohypertrophy of calves that is actually connective and fat tissue that is one well-known sign of dystrophinopathy. Sometimes early in the disease course, boys can complain of pain in their muscles. And as I said earlier, disease is progressive, and patients lose their ability to walk at the age of 12, most of them. Some can walk a bit longer, and some of them lose the ability earlier. When they stop walking, they develop, almost all of them, neuromuscular scoliosis, and scoliosis is one of the causes for deterioration of lung function. Laboratory signs can be present or are present since birth, and the well-known biomarker of dystrophinopathies is creatine kinase, CK, and its levels are pretty high in dystrophinopathies, the highest in Duchenne muscular dystrophy, and as the disease progresses, they become lower during the years. There are also increased levels of liver enzymes, aminotransferases, but they are not actually from the liver, they are muscle enzymes. And this is sometimes patients with Duchenne dystrophy early in the course of the disease are being treated or diagnosed as liver patients, but that is not the case. Cardiomyopathy is also can be present in patients with DMT. It develops later, a bit later during the course. So 1/2 of the boys have cardiomyopathy at the age of 18 years. It is not actually symptomatic till the late stages of the disease. So heart ultrasound should be done in patients with Duchenne, but also boys can have conduction disorders. So ECG is also suggested. During the course of disease, there are multiple complications, and the important one, all are important, but maybe more important ones are orthopedic complications like contractures and scoliosis, but also bone fractures. If a patient has bone fracture, it can make him to lose his ambulation much early. So bone health is one of the topics that has to be cared in DMD. Pulmonary function deteriorates, and at the age of 16, usually vital capacity is less than 50%. And when that is the case, that can lead to acute respiratory failure during respiratory illnesses. Some patients with Duchenne have cognitive functions affected and have behavioral disturbances, even some cognitive deficit and psychomotor development delay. Those are actually characteristics of DMD. Boys can have autism spectrum disorder, problems with attention and hyperactivity, anxiety, but some patients have normal IQ and even above the average. So that is not the rule for all, but sometimes it can suggest disease in young boys, cognitive problems. This is a diagram that shows disease progression. There are a few phases, and the first one is called pre-symptomatic stage. Nowadays, we are developing new therapies, so it is expected that Duchenne should be diagnosed early. So developmental delay, positive family history, or elevated CK should raise concern about possible dystrophinopathies. So as you can see in the beginning, boys can improve due to psychomotor development, but then they reach a certain plateau, and then the disease causes muscle decline, and they in the end become non-ambulatory. To establish the diagnosis, nowadays, one should try to have molecular diagnosis or gene analysis positive, and it is not always possible. So muscle biopsy still has its role in diagnosis of DMD, but that is really, really rare nowadays. So what should raise concern that there is possibility of DMD? It is delayed motor development in a patient with positive family history, or actually any problems with the muscles, such as head control, difficulty running, jumping, frequent falls, muscle pains, myoglobinuria, or some cognitive problems, speech, learning, behavioral disturbances. In a patient with positive family history, those signs should be indication for gene testing. Otherwise, if there is delayed psychomotor development, especially if a child doesn't walk till the age of 16 or 18 months, or have clear signs of proximal muscle weakness. Also, unexplained elevation of aminotransferases should be a sign to check CK. And actually, CK is elevated in 100% of patients with DMD, and it is also elevated in 100% of patients with Becker muscular dystrophy. So if CK is normal, then you can rule out dystrophinopathy. And the diagnostic process is, if you have a suspicion that I explained earlier, you should test for CK. If CK is elevated, then gene testing for deletion or duplication. This test will cover a majority of mutations, and it is a cheaper test, and I think it is done widely in lots of institutions. If this test is negative, we still didn't rule out Duchenne, then genetic sequencing is indicated. And if that is negative, then this is the reason for muscle biopsy, because there might be some intronic mutations or other that are not present in the gene. But testing for deletion of exons or duplications will cover majority of mutations. Treatment of the Duchenne, it is not like in some other disorders like SMA that we will hear lately, but there is a significant progress in the last years, but still there is no cure for dystrophinopathies. Treatment is multidisciplinary, consists of pharmacotherapy, physical therapy, surgical treatment, and other therapy. I think multidisciplinary approach is still a problem for some of us, and it is not always easy to have a real multidisciplinary team that will be functional, and especially that will be patient oriented. Not just patient oriented, but multidisciplinary approach should be appropriate for the certain stage of the disease. And as you can see here, actually lots of specialists and lots of medical workers should be involved in multidisciplinary team for Duchenne muscular dystrophy. And this is actually nicely explained in standards of care for patients with DMD, and there is a really a wide range of experts that should take care. And as I said before, we should try to be patient-oriented. And now I will say a few words just about available pharmacotherapy. Nowadays, corticosteroids are most important therapy in patients with DMD. They should be started before the significant decline in muscle function that is usually at the age of 4 to 5 years and never before the age of 2 because side effects of corticosteroids. And it has been shown that corticosteroids really do improve muscle strength and muscle function, and they prolong a bit ambulatory phase, but they also not improve but slow deterioration of pulmonary function, and they can delay the development of cardiomyopathy. Usually 2 corticosteroids are used, it's prednisone and deflazacort, and there are still some discussions about the regimen of taking the steroids, and it is still not fully known what is their exact mechanism of action in Duchenne muscular dystrophy. Nowadays in Europe, we have a drug that is used in patients with nonsense mutation. It is point mutation that causes premature stop codon in DMD gene. This mutation is present in 10 to 15% of boys with DMD, and the drugs works on ribosomes to skip the premature stop codon. It has been shown in clinical trials that it slows deterioration of results on 6-minute walking tests only in certain group of patients. But as it can be shown later, it really helps a bit in the loss of ambulation. It prolongs the time that the patient is ambulatory. And more than that, it lowers deterioration of functional tests in patients with DMD. Ataluren is registered in Europe and not registered by FDA. Another group of the drugs that exists are antisense oligonucleotides used for exon skipping. The idea is if you have out-of-frame mutation and there is possibility to skip some exon nearby to make out-of-frame mutation to become in-frame mutation and theoretically to change Duchenne phenotype to Becker. So this is the idea, and it is applicable only for certain mutations. You have to check if a deletion is applicable for certain exon skipping medication. And it has been shown that it really do improve dystrophin expression in muscles, but still it is from, for example, from 0.2 to 0.6% of normal dystrophin. And because of that, European agency didn't approve application of exon skipping therapy, and it is improved by FDA, but mostly due to dystrophin expression and not always due to clinical results. Besides eteplirsen, there are also other exon skipping drugs that are used for skipping other exons. And finally, I do hope that we will see this in future, maybe near future, it is gene therapy for Duchenne muscular dystrophy, but there is still a problem. As I said, DMD gene is a huge one, and it is too big to be put whole in a viral vector. So the idea is to use a shorter variants called mini dystrophin or micro dystrophin to introduce a missing gene to a patient. There are actually few clinical trials, and the drug is still, all of them had some problems, but I think we can expect that it will happen in the future, but still gene therapy is not yet in clinical practice. There is also possible, and it is there are some clinical trials of cell therapy, especially for cardiomyopathy, and some other drugs are being tested. Some of them work. Some clinical trials are stopped. Vamorolone is corticosteroid that is used because of less side effects, and some other drugs that actually work on muscle and try to prevent their deterioration. So in the end, we have some improvement in therapy of Duchenne muscular dystrophy. It is a really severe disorder that affects boys, the most frequent muscular dystrophy. We will have to try to have good expression of dystrophin in future therapies. It is expected that 30 to 50% of dystrophin in the muscle should be enough for a patient to have less symptoms. Today, we still cannot achieve that. And today, we have some therapeutic options that we didn't have a few years ago. So thank you very much for your attention. I tried to give you an overview of present situation with Duchenne muscular dystrophy. And thank you once again, and greetings from Zagreb, Croatia. This is our Christmas market, and if you are able, you are all invited to visit us. We are very proud of this, and I wish you all happy holidays. Thank you once again.
Thank you very much for this overview, but also for your invitation to Zagreb. It's very tempting. I would like now to open the floor for the questions or comments from the public. Please just open your microphone and ask the question.
Maybe just one comment. Hi, Dorica. Very nice talk, Ivan. And as you mentioned, there are questions and issues around gene therapy for Duchenne due to the fact that there are different microdystrophin which could be expressed, which could have or generate some responses, immune responses from patients. So I just wanted to highlight the fact that most of the clinical trials trying to express microdystrophin unfortunately had adverse effects for a few patients. But the very, very positive, and that's really the message I want to carry, that's a positive message, is that whereas it never happened before for any pathology, all the competing companies came together to share their information about this adverse effect, to try to understand together how to improve their gene therapy treatment and how to overcome these side effects and increase the safety of treatment for patients. And so I think that's unique because before, usually these pharmaceutical companies were just competing and not sharing their information, particularly when it's about issues. They prefer to stay silent. And so just to say that one of the reasons why they accepted was because there were pressure from patient organization for them to share their data. So first, to let you know about this change in the pharmaceutical company way of looking at clinical trial, they understand now that for rare disease, they need to pull the data together to be able to understand what is going on. And second, that patients were really instrumental in convincing them to share this data. So even if you do not, fund the clinical trial or fund research, you can have a very important role to make sure that the one who are actually organizing this clinical trial are sharing the data and make sure that it increases the safety and efficiency of the treatment for all patients. So that was my message.
Thank you.
Thank you very much. If you want to intervene, Dr. Ivan.
No, thank you.
Okay. Okay, if there are other comments or questions, Arabella?
Yeah, I actually do have a question. First of all, I'm very happy to see you again, Dr. Lehmann. We had the pleasure of meeting in Ljubljana in September. Yeah, so and I was happy to see that you are very engaged with patient organizations as well. So I don't have a very medical question, but I just want to say, because your specialty is actually children with Duchenne. So you have a lot of engagement with children and with children's parents and caregivers. Do you think, how do you see these interaction with the families and with the caregivers? Because I know I'm a mother of 2 children with CMP, not the same, but still with a disability. So I know for parents and for caregivers, this can be very challenging. And for us, it is so important if we find those kind of doctors that are very supportive and very understanding. And I really think that you are that kind of person. So do you have a special message for the families of children with Duchenne? Something that can actually raise their optimism, I don't know, encourage them? Because you are actually close to families, but also with patient organizations.
Yes, thank you for the questions. I'm not sure if I'm an example for that, but I think that first of all, our role is to try to establish the best medical care for the patient. And I see that in my hospital and in our referral center, we still struggle with that, especially establishing multidisciplinary teams. So I think this is our first role as a physicians, to try to be good or the best in medical care, and we still have problems with that. What I can offer, I can't offer much. It is always good idea to listen to needs of patients, and sometimes, not sometimes, but patient organizations are the ones that can channel those needs, and that can put them in format, so you know what are the needs of the patients. And another role of the doctor is, I think, that one should be available for the patients, especially if something happens and if there are extra needs. It would be nice to have the opportunity to form excellent teams, to have some teams around the country, but I think it is maybe too big task. It's important to have a team, really good team at first at our hospital or in one's hospital, and then to improve the standards of care. So that is my opinion regarding that. And dealing with parents, it's I'm a pediatrician, so yes, it is difficult sometimes. You are right, especially in severe diseases like Duchenne muscular dystrophy, and sometimes you have to engage parents. Some parents are really educated and they know about the disorder and about their needs even more than we do sometimes, but sometimes you have to try to engage the caregivers more. That is also my point of view. Thank you. I hope I mentioned something that you wanted to hear.
Thank you very much. Are there any other questions or remarks, comments? Thank you very much. And we'll go to the next presentation if we don't have any other questions. And I will invite Dr. Anna Macias, which is a neurologist working in the Department of Cardiology and Neurology Medical University in Warsaw. She is going to speak about advances in clinical trials in non-Duchenne muscular dystrophy. Thank you very much for being today with us, and the floor is yours.
Hello, thank you for invitation. Hello from snowy Poland. I hope that you can hear me, and I would like to put on the presentation now. Okay. Yeah, can you see the screen now? Yes? Okay.
Yes, we can see.
Okay. I would like to briefly present the advances in clinical trials in non-Duchenne muscular dystrophies. Why non-Duchenne? Because presenting all the clinical trials in Duchenne muscular dystrophies will take the whole lecture, yeah, another lecture. I'm happy that Dr. Lehmann already presented briefly the state of clinical trials in Duchenne muscular dystrophy. Of course, Duchenne muscular dystrophy is the most frequent one, but therefore I would like today to focus on all the other ones. Okay, first of all, this is my disclosure. The source of information I based on generally available information, like, for example, the site clinical trials and trial sponsors publications. I will limit, due to the limited time, only to pharmacological trials, only to drug trials. First of all, I am a clinician, and I am not currently engaged in any of the presented clinical trials. I just did this review predominantly to present it to the patients community, yeah, to have the information gathered in one place for them. We can divide muscular dystrophies into different types. First are dystrophinopathies. These include Duchenne and Becker muscular dystrophy, limb-girdle muscular dystrophies, which overlap in some percentage with congenital muscular dystrophies. Another type is facioscapulohumeral muscular dystrophy, myotonic dystrophies type 1 and 2, Emery-Dreifuss muscular dystrophy, and oculopharyngeal muscular dystrophy. And currently, interventional clinical trials, pharmacological trials are going in the of these therapies, which I highlight as well, but I will skip limb-girdle muscular dystrophies. And I will talk today about these types of dystrophies. First of all, we already know that steroid can improve the patient's condition, can slow the progress of the disease in Duchenne muscular dystrophy, but we still don't have any guidelines concerning other types of muscular dystrophies, whether it is beneficial to patients to use steroids in other types of muscular dystrophies. So there are some trials which are ongoing in Becker muscular dystrophies and limb-girdle muscular dystrophies. In Chicago, in USA, there is a trial of weekly prednisone dosing. This is ongoing. This study has not published results yet. There was a study of deflazacort use in limb-girdle dystrophy type 1. However, it was terminated early, unfortunately, because this is one of the dystrophies which, according to some data, could benefit from the steroid use. It was terminated early due to high dropout, unfortunately. There is another study going on in Becker muscular dystrophy with vamorolone. This is currently recruiting the patient in USA. Vamorolone is called a dissociative steroid. It is a steroid derivative which has retained anti-inflammatory activity, but doesn't have GRA-related transactivations, so doesn't have many corticosteroid side effects. For example, does not present mineralocorticoid activity. Yeah, so this is the safest alternative, and it should retain the anti-inflammatory activity of steroids. Okay. Apart from the trials with steroids, there are some very interesting new substances tested. One of them is EDGG 5506 by Edgewise Therapeutics. It is a selective fast skeletal muscle myosin inhibitor. Myosin, it is a protein which is present in the muscle and which is responsible for the contraction of the muscle. And fast fibers, we have fast and slow myosin types, and fast fibers are more susceptible to injury due to muscle contraction related stress. This injury due to muscle contraction related stress is especially important, is especially enhanced in muscular dystrophies, in different types of muscular dystrophies. And in mouse dystrophinopathy model, selective inhibition of fast fiber contractions prevents this type of injury. So the sponsor designed a trial in which the substance is blocking the fast skeletal muscle myosin. And there was a preliminary study called ARCH in patients with Becker muscular dystrophy. This study is still ongoing, but has the interim data after 6 months. And we can see this is the North Star ambulatory scale, which assess the ambulation. The higher, the better score. And this is the natural Becker muscular dystrophy history, and this is the score, the progress of the score in the Becker muscular dystrophy patients treated by this EDGG 5506. And this is the creatine kinase level, which is decreased by about 40% after 6 months. So these are very encouraging preliminary data, and the company has started already 2 other trials. Canyon trial in adults and adolescents with Becker muscular dystrophy, which is currently recruiting patients. Lynx trial in children with Duchenne muscular dystrophy. And another very interesting trial is planned in patients with Becker, but also limb-girdle muscular dystrophy, 2I, this is type R9 currently, McArdle myopathy. Yeah, so this is planned soon to start. Another group of clinical trials are trials with gene therapies, and I collected them in form of the table. There were some trials of gene therapy in dyspharinopathy, in limb-girdle dystrophy type 2. The very preliminary phase 1, this trial is completed, but the results are still pending. We don't know the results. There was also a study in this sarcoglycanopathy type alpha, LGMDR type 3, which also has been completed, and it showed the increased sarcoglycan production and local muscle function improvement in the injected limb. This was a single limb injection. Another trials are currently active is gene therapy in sarcoglycanopathy beta, LGMDR4, which is also a preliminary phase clinical trial is ongoing but not recruiting patients. And now a very interesting clinical trial with gene therapy in limb-girdle muscular dystrophy type R9, fukutin-related protein related. This is quite a common type of limb-girdle muscular dystrophy. And currently the study is recruiting patients in Denmark, France, and then UK. Another very similar study is planned, but not yet recruiting with another preparation, another pharmaceutical company. Okay, so this is these are these preliminary results of the sarcoglycan gene therapy study, which demonstrate no sarcoglycan expression before the therapy and sarcoglycan expression after the gene injection. Okay. Apart from gene therapies, there is another, a very simple particle, very simple substance, which is tested in limb-girdle muscular dystrophy type 9, R9. This is ribitol. Ribitol, this is a very simple chemical substance, which is a sugar alcohol formed by the reduction of ribose, and it is a substrate for dystroglycan glycosylation by FKRP. This is the enzyme which is mutated in this type of limb-girdle muscular dystrophy. Dystroglycan is present in the muscle cell membrane, and it is very highly glycosylated. It means that there are sugars which are connected to the protein core, and one of the enzymes which connects these sugars is fukutin-related protein. These sugars connect the dystroglycan and muscle cell membrane to the extracellular matrix, to the laminin here. And in normal situation, when FKRP is not mutated, it connects the ribitol to the alpha dystroglycan, and this glycosylates alpha dystroglycan and connects it to laminin. Mutant FKRP is unable to do it. This activity is only trace here, and only some ribitol particles are incorporated into the glycosylation process. But in a situation of exogenous ribitol excess, this mutated FKRP protein, this mutated enzyme, which has an excess of the substrates, starts to work better, more efficiently, and glycosylates alpha dystroglycan more efficiently. This was proved in preclinical models, and now clinical trials have started. Phase 2 trial is ongoing, not recruiting the patients currently, but it has very encouraging preliminary results as well. This is the dystroglycan glycosylation level pre-therapy and post-therapy, day 19. And we can see that the therapy is the reduction of the creatine kinase level. Okay, sorry, I must. This is these are the results of 10-minute walking tests. Phase 3 study is planned soon, possibly this year. Another type of muscular dystrophy, facioscapulohumeral dystrophy, Here we have also some very promising preliminary data. A very interesting drug which is tested is losmapimod by Fulcrum Therapeutics. It is an inhibitor of P38 MAP kinase. This inhibitor reduced DUX activity in preclinical models. DUX, it is a gene which is overactive in the facioscapulohumeral dystrophy. So a phase 2 study, REDUX-4, was constructed to show whether this losmapimod reduces DUX and improves the clinical parameters in patients with facioscapulohumeral dystrophy. Unfortunately, the results show no DUX4 reduction in the patients, but better functional outcomes. For example, this is the reachable workspace. This is the distance, the space which the patient can reach by his hand, because this kind of muscular dystrophy predominantly affects upper limbs. Yeah, so this reachable workspace was better in patients treated by the drug than with placebo. Muscle strength tested by dynamometry was also better in patients treated with the drug. And now there is a phase 3 study called REACH, which is recruiting the patients not only in the USA, but also there are some centers in Europe which are involved in this study. And I hope that the results will be as optimistic as the preliminary results. Another trials in facioscapulohumeral dystrophy are trials with creatine monohydrate. This is a dietary supplement with anti-myostatin antibody. Let's hope that this will give promising results, because up to now, anti-myostatin antibody, we had great hopes, but the results were disappointing a bit in Duchenne dystrophy, for example. And trial with antioxidants used in Montpellier in France. And the last type of muscular dystrophy, which I would like to present, is myotonic dystrophy. It is also a very common type of muscular dystrophy, the most common one in adult patients worldwide. In this type of muscular dystrophy, the group of drugs which are of particular interest lately are antibody-bound antisense oligonucleotides or small inhibitory RNA, which are blocking the DMP kinase. DMP kinase, DMPK, is a protein which is again overactive, and it's not overactive, but it forms a large amount of mutated abnormal mRNA, which is toxic for in the long term for the cell, which affects splicing of other proteins. So this abnormal DMPK is blocked by these oligonucleotides or small inhibitory RNA. And there are two clinical trials by different pharma companies. First is Marina study with siRNA antibody conjugated, and the other one is ACHIEVE trial, which is now recruiting the patient, but only in New Zealand. Very similar mechanism of action, and we will see whether the results will reflect the very encouraging preclinical data for these two substances. Another drug which was tested in a very specific type of myotonic dystrophy, namely congenital type, a very severe form of myotonic dystrophy, congenital or juvenile type, is tideglutide. Tideglutide, it is an inhibitor of glycogen synthase kinase 3 beta. And the preliminary study found clinical benefit in the majority of subjects after 12 weeks of treatment. The results have been already published, and now there is a study called REACH, CDM, REACH, Congenital Dystrophy, Myotonic Dystrophy, which is recruiting the patients in some countries, unfortunately not in Europe yet. So as you can see, there is much going on in the clinical trials in different muscular dystrophies, not only in Duchenne one, which is I think it is a very optimistic for the future. In the contrary to the last 5-10 years, when we didn't have any option to these patients with muscular dystrophies. I would like finally to mention unfortunately, some most important negative clinical trials. Anti-myostatin antibody, as I already mentioned, disappointed us a bit. It was tested, for example, in LGMD-R9, FKRP-related muscular dystrophy, with no improvement. And moreover, there were some doubts about whether it may be impaired cardiac function. And another negative study, this is an old one, was deflazacort in dysferrin-related limb-girdle muscular dystrophy, which, in contrast to what was expected, caused decrease of muscle strength when the patient took the tested drug. And another drug which was which raised high hopes a few years ago. It is Resolaris. My patients in Poland always ask me about this drug. Is it in the market already? I don't know. After some promising phase 1-2 data, these are the number of clinical trials, there were plenty of them. This drug was granted fast track designation and orphan drug designation in 1915-1917, but then it simply disappeared from the company's pipeline, and probably there are no new trials with this drug. Okay, so thank you very much. I hope that this brief review was interesting for the patients and doctor community. And I would like to wish you all happy holiday season. And again, thank you for inviting me.
Thank you very much for this presentation. And I would like to open the floor for questions and comments.
Hello, hi. I have a question about the very last drug. When the company presents at any kind of conferences, do you, as researchers, ask about what happened to this, why the results are not published, why has it disappeared, and why there's no information for the patient community?
This is a very difficult question, because this company now in her pipeline does not have any drug tested for any neuromuscular condition. Yeah, they focused on other clinical areas. So we do not have contact with the company representatives in the conferences. Yes, I am puzzled what happened to this drug, because the first clinical data were quite optimistic. Although there were some adverse effects, and maybe that's it. Okay.
Thank you.
Other questions, comments? If not, I would like to continue, and the next presentation, sorry, the next presentation will be about SMA, the introduction of therapies for spinal muscular atrophy has changed the clinical landscape, transforming SMA from a lethal to a treatable disease. Our next speaker, Milos Bruksanin, sorry if I spelling your name wrong, is working at the Center for Human Molecular Genetics, Faculty of Biology, University of Belgrade. He is the chair of biochemistry and molecular biology. He will speak about SMA from a fatal to a treatable disease. Milos, the floor is yours, and thank you very much for being today with us.
Good afternoon to everybody, and thank you very much for the invitation. First of all, I must say that my connection right now is not very stable. So if there are any problems during the presentation, please give me a signal so that I can at least turn my camera off. Thank you for this introduction. I work at the Center for Human Molecular Genetics, which belongs to the Faculty of Biology in Belgrade. For the past 25 years, the center has been focused on diagnostics and research of rare neuromuscular diseases. And I myself have been involved in research on spinal muscular atrophy for more than a decade now. And currently, I am the project leader of the newborn screening pilot project in Serbia, and I also work on the identification of biomarkers for treatment response to genetic therapies in SMA. We usually start every lecture on SMA with this horrifying sentence that the SMA is the most common genetic cause of death in children, with the average incidence of 1 in 6,000 to 1 in 8,000 children. But the question that I will ask and try to answer is, can we make this phrase a thing of the past? So I will briefly discuss how things used to be in the past. So unfortunately, for decades, individuals living with SMA and their families did not have any hope. What does this mean? We would perform genetic testing for a child, confirm the diagnosis, and then the doctor would usually tell the parents to bring the child back home, to love them, take a good care of them, but also to witness the inevitably progressive natural course of the disease. Of course, some standards of care and support were available to these individuals, such as nutritional care, pulmonary care, physical therapy, or palliative care. And indeed, these standards of care were affecting positively the overall quality of SMA individuals. But however, they were not able to remove the initial cause of the disease. The only prevention that we were able to offer to the affected families were carrier testing, first of all, to see whether other family members are carriers of the disease-causing mutation. And if they were found to be carriers, then prenatal testing was available and offered in each future pregnancy. So genetic testing in the affected families definitely brought immediate benefit for affected families. And in many disease-affected families, this was the only opportunity and way for having healthy offspring. However, in the past several years, we have witnessed the approval of several therapies for SMA, these therapies showed great potential in both preclinical and clinical studies. One of the advantages of these therapies is that they are genetically designed, which means that they directly target the disease cause. All approved therapies for SMA showed very high therapeutic potential, but only if they are applied before the onset of symptoms, which means early after birth in the so-called pre-symptomatic stage. And this is precisely the reason why we today talk so much about newborn screening as a way of disease prevention. And compared to carrier screening and prenatal testing, which used to be offered only to disease-affected families, newborn screening is offered to every family and should be available for each newborn, bringing equal chance for every baby to be diagnosed and effectively treated on time. SMA is marked by premature and irreversible death of motor neurons in the anterior horns of the spinal cord. So in this photo, you may see that the arrow tips show very thin anterior horns and that the entire arrows point towards normal posterior horns. So these anterior horns are very thin because of the death of motor neurons. What is very important to understand is that in SMA individuals, almost 95% of all motor neurons are completely lost within first several months of life, which as a consequence leads to the muscle weakness and atrophy. And unfortunately, if no treatment is applied, then the disease most frequently has the fatal outcome. Approximately 60% of all SMA individuals develop the most severe type of the disease, which means that they present symptoms very early after birth. They never have the ability to sit alone, and very rapidly after birth, they lose the ability to eat and breathe without any support. Other forms of the disease are less severe, we say milder, but they also carry permanent disability and invalidity. So why do motor neurons die prematurely? The answer is simple, because there is a lack of a protein which is vital for their survival and normal function, and this protein is called the SMN protein. In the DNA molecule of a healthy individual, the information for the production of the SMN protein is contained within a gene which is called the SMN1 gene. The SMN1 gene is the main source of production of the SMN protein, because the SMN1 gene always produces normal functional SMN protein. But however, in a healthy individual, there is also the SMN2 gene, which is called the backup gene. It is only partially functional, and it produces only about 10% of the normal SMN protein. The situation is different in an SMA individual, where there is no functional SMN1 gene, and unfortunately, the only source of the normal SMN protein is the SMN2 or the backup gene. And these 10% of the protein that is produced from this backup gene is not sufficient to prevent the onset of the symptoms. So if we know that in an SMA individual, there is no functional SMN1 gene, but there is the backup SMN2 gene, which produces low and insufficient amounts of the normal SMN protein, we may review the available therapeutic options for SMA. One of the therapies that has been approved is gene therapy, or Zolgensma, which actually represents the introduction of the functional SMN1 gene into the body through one-time intravenous infusion. And this is also one of the most expensive therapies in the world. Another approach to use is to target the SMN2 gene in such a way to make it produce more functional SMN protein and not only 10%. One such therapeutic is nusinersen, which is an antisense oligonucleotide. In other words, it is a molecule which cannot cross the blood-brain barrier. It cannot enter the central nervous system where its action is needed. And for this reason, this therapeutic has to be injected directly into the spine, or as we say, via intrathecal injection. Another way to achieve the same is to use a small molecule, risdiplam, which is taken orally, and it is important to understand that both nusinersen and risdiplam are lifelong treatments. So the situation seems quite simple. We have the disease, we have 3 available therapeutic options, we use the therapies, and we cure the disease. But unfortunately, this is not the case in spinal muscular atrophy. So unfortunately, individuals with SMA are diagnosed with a significant time delay after the onset of their symptoms. The patients with the most severe type of the disease are diagnosed with a delay of 7 or more weeks, while individuals with milder forms of the disease are diagnosed with even longer delay, sometimes up to 1 or 2 years. This diagnostic delay reduces therapeutic efficacy, but it is also fatal in the most severe type of the disease. Why? It seems that these 7 or more weeks is not a long period, but however, I must remind you that in these 7 weeks, almost 95% of all motor neurons are completely lost. And for this reason, such child has completely and irreversibly lost any possibility to sit unsupported, to eat or breathe without support, and even with the therapy comes too late, and the disease unfortunately has the most tragic outcome. So what I would like to highlight here is that in the era of innovative genetic therapies, when extremely high financial funds are spent for treatment of SMA individuals, diagnostic delay represents the major obstacle in achieving the maximum therapeutic benefit. And once again, I would like to highlight the importance of early diagnosis and early treatment. So the reply to my question from the beginning, can we make SMA a treatable disease? Yes, we can, but through the newborn screening. So this is a map from last year presenting different countries of the world and the availability of the treatments for SMA and the availability of the newborn screening. In this map from last year, Serbia has been recognized as a country where treatment options are available, but with no screening implemented. So now I will briefly share our experience to show you how we managed to make SMA a treatable disease in our country. So first of all, what treatments are available in Serbia? The first therapy for SMA came into our country in July 2018 when nusinersen applications started. Up to the present moment, if I'm well informed, we have a total of 35 persons with SMA treated with nusinersen, including 4 adult type 3 patients. Almost at the same time, 8 SMA persons received risdiplam as a part of the clinical study, and up to the present moment, totally 30 of them have received this treatment. Both nusinersen and risdiplam are financially covered by our health insurance fund. Regarding gene therapy, we have 9 SMA patients who received this treatment up to the present moment, and they had all previously been treated with nusinersen. However, until recently, gene therapy was not registered and approved for use in Serbia. And still, it is not financially covered by our health insurance fund. So how did these patients receive this expensive treatment? Through numerous fundraising campaigns. But one of the advantages of these fundraising campaigns is that they enabled raising of the awareness of SMA in our country and the awareness of early intervention. So, bearing in mind that therapies are available in Serbia, and with the emphasis in SMA being placed on early intervention and early diagnosis, we knew even then in 2018 that the newborn screening was the next logical step we had to work on. So for this purpose, in 2018, in the Center for Human Molecular Genetics, we optimized and validated a commercial test for the newborn screening, which used dry blood spots as the initial sample. This technique was shown to be 100% specific and sensitive in detecting SMA individuals with no SMN1 gene. The entire laboratory workflow takes about 4 hours for the analysis of 91 samples. And according to the Statistical Office of the Republic of Serbia, there are on average 180 babies born daily in Serbia, which in other words means that for all the babies born on the same day, the screening can be performed within a single day. So what was our path towards the newborn screening as a way of disease prevention? First of all, in close collaboration with the association SMA Serbia And our clinicians at the University Children's Hospital, we prepared a project proposal, which we sent to the Ministry of Health. Unfortunately, our project proposal was rejected without any explanation, which was very disappointing for us at this moment, because it made us realize that a huge experience that we had, that the close collaboration, a good idea, that actually this was not a guarantee of success. But nevertheless, we didn't lose hope. And in September last year, the association SMA Serbia organized a roundtable on SMA, where we tried once again to convince the government that the early diagnosis was extremely important. However, they did not understand this, or at least they didn't want to understand it. Luckily, there were representatives of the pharmaceutical companies present in the audience, and they recognized this urgent need to screen for SMA early after birth, and they agreed to provide complete financial support for a one-year pilot project. So in order to establish the newborn screening in Serbia, we decided to start from one maternity hospital, which is one of the biggest maternity hospitals in the entire country, and it is located in Belgrade. And in this hospital, around 8,500 newborns are born annually. So we obtained the ethical approval from the hospital's ethical boards, and upon giving birth, every mom is asked to sign an informed consent if she wants her newborn to be included in the SMA screening. If she agrees, then 2 drops of blood are collected from the heel of the newborn and saved on a specific card, which is adapted for genetic analysis. Then the following day, the samples are transported to the Faculty of Biology, where they are specifically tested for SMA. So, as I have already said, the awareness of SMA and of the importance of early diagnosis was already present in the general public in Serbia due to numerous fundraising campaigns for collecting the funds for gene therapy. However, when this project started, we as project participants tried to spread the main message and the impact of the project to the general public through numerous media appearances. We set some objectives that we wanted to achieve. First of all, we wanted to create organized and coordinated work at different levels of healthcare, which means that we wanted to establish the entire process to work successfully, from the sample taking until the approval of therapy and patient follow-up. Of course, the ultimate goal was to prepare the ground for the establishment of the newborn screening at the entire territory of Serbia. And these steps were in accordance with the National Rare Disease Program, which discusses improvement of the availability of the newborn screening and therapies for rare diseases. However, on the right side, you may see some of the challenges that we faced. First of all, in the beginning of the project, many parents refused to participate. They didn't want their newborn to be tested for SMA at birth, usually due to the fear of the unknown or due to the parental anxiety immediately after childbirth. Another thing, we were not able to foresee whether the country would have enough funds for possible positive cases that we may identify in this project. And the main obstacle was, of course, obtaining the government support for the spreading of the screening at the national level. Our pilot project started in April this year, and until today, we have tested approximately 5,000 newborns. Since we included only one maternity hospital, we expected to detect maybe one or maybe none newborns with SMA, but actually very quickly we identified 2 positive cases in our pilot projects. And we identified them very quickly because we confirmed their diagnosis when they were approximately 6 days old. So then the therapeutic options were discussed with the family, and the therapies were approved within 10 days. And for both newborns, the therapies were given when they were less than 1 month old. So the total number of therapies that we approved based on the results of our newborn screening pilot project is totally 3. Why do I say 3? Because we identified 2 newborns, and the first newborn has an older brother who was also confirmed to have SMA, even though he was completely asymptomatic at the age of 16 months. So now all 3 children are given the therapy. During this 8-month period, we established a complete screening and diagnostic algorithm, which means that we now have the way to confirm each positive newborn screening result, to establish the pathway to determine the SMN2 copy number, to discuss the treatment. But what is interesting, we insisted on pre-symptomatic initiation of treatment in all individuals, including those with 4 SMN2 copies, which are predicted to develop milder forms of the disease. Why did we insist and demand immediate treatment for all of them? Because this way we can reduce or completely prevent the irreversible motor neuron damage, and thus provide completely normal or almost completely normal life for these babies. So what are our achievements? We created the organized and coordinated work at different levels of healthcare. We prepared the ground for spreading of the newborn screening, but still we are not ready for its implementation at the entire country level. Also, we achieved higher rates of participation in the project because now almost every family accepts to test their newborn for SMA. And we also realized that parents are able to rationally cope with the unexpected SMA diagnosis in their newborn. And we are just about to start a new round of conversations with the government, with the Ministry of Health and the Health Insurance Fund, where we will try to present the challenges and the strategy to implement the screening at the entire country level. And we hope we will be able to do so from the beginning of 2024. So these are some very important take-home messages. First of all, SMA is a rapidly progressing, devastating disease, which, as I said, is caused by the premature and irreversible loss of motor neurons. This is the reason why early diagnosis and early therapy application is essential in SMA, because only this way we can achieve the maximum therapeutic benefit. So regarding my question from the beginning, can we make SMA a treatable disease? I will say once again, yes, we can, through newborn screening, because newborn screening accurately identifies babies with SMA at an early stage before the onset of symptoms, thus giving us the chance to treat the baby in the perfect moment and to make this baby live a completely normal or almost completely normal life. Of course, the SMA community strongly supports and advocates widespread implementation of newborn screening. And only by our joint efforts, we can make this most common genetic cause of death in children a thing of the past. So in the end, I would like to thank you for the invitation once again and for your attention. This is a photo of me carrying a baby, the first newborn that was diagnosed with SMA in our pilot project when she was only 5 days old, and she received the therapy when she was 17 days old. I really believe that we saved one life, and I am absolutely sure that this baby will never develop any symptoms of SMA. Thank you very much once again.
Thank you very much for this presentation. It was a great overview of the disease, but also of your stepwise approach in Serbia in getting to a newborn screening for SMA. That's wonderful how you approach this, and this is a great example for all of us. And now I'd like to open the floor for questions. I'm sure you have many questions.
There are some raised hands, so maybe they can just open the microphone or I don't know, Dorica, you name them. I see Ipeg and Liv, you have some questions.
So, I start. Okay. Thank you, Milos. It was a great presentation. Thanks a lot. I have two questions. In Turkey, we have only nusinersen treatment for the time being. And also we have some criteria to keep the treatment, because you need to give the treatment in each 4 months. But our government looks to some criteria, for example, increasing the Hammersmith scale and other ones. My first question is, do you have any criteria to continue nusinersen treatment? And the other question, since it is a continuous treatment, how do you manage this timely application of each doses? Because in Turkey, we have to track the process by families. They have to go to the doctor and track the right timing. So we lose time.
Okay, so the first question was, of course, the situation is the same in Serbia. Since our health insurance fund provides financial support for the therapies, they need to see that the patient shows an improvement, which is very difficult because they want to see that a patient is a responder. So what is a responder? It is very an unusual question. Is it something that we can measure on a scale? Maybe a clinician would say, yes, the patient improved for 1 point, but is it significant or not? Another thing is maybe the patient, if the patient is old enough, can say, I have more strength, I have a better quality of life, then it means that he responds to therapy. Maybe it's the molecular biologist who should say, yes, we can see that the neurofilament levels decreased, so this patient is obviously a responder. So we have the same problem, how to prove that the patient responds to therapy. I think that our clinicians have to write a report every year for the National Fund, and that they follow the motor scales. But I am not sure that our fund is so strict. So if you describe any kind of improvement in a patient, they just keep on providing the funds for the therapy. So once that you get into this system of getting the funds for the therapy, if I am well informed, in order to receive the funds each year, our fund is not so strict. So you just have to show them that there is any kind of an improvement. So your second question was, how do we manage to keep parents coming, if I understood well, at the exact time when they are supposed to come to receive the treatment.
Yes, timely management of the ongoing treatment, I am asking, because normally in Turkey, we have to track these timings and go to the several doctors before each dose of nusinersen. But maybe in your country, the doctors or the health system manage this timely application of the new doses.
We do not have any very effective system. The thing is that Serbia is a very small country, and that patients are treated in Belgrade. So it is centralized, and there are not many patients. Each of them have their own schedule. Each of them has their own clinician at a different institution. So I think that in Belgrade we have 4 institutions where these individuals are treated. So it's not very difficult to organize a visit at the exact date when they are supposed to be treated. Sometimes it happens that due to some other health complications, the dosing has to be postponed for a few days. But I think it's something like to be expected. So we do not have any very effective system. The thing is that the country is small, that all patients come to only several institutions. And I'm saying this because in Serbia, we have around 65 babies born per year. And I know that there is 1 million babies born in Turkey. So I'm making this comparison just to let you know why things are maybe easier in our country than in Turkey.
Yes, yes. Thank you so much. It's great, and congratulations for the newborn screening. All you
Thank
made
you very
was
much.
great. Thank you very much.
Thank you.
Are there any other questions, comments?
Yes, can I make a comment? I am the second one from Turkey. Also, we have started dominating after Epicana. My name is Onur Çakır. I am from Turkey as well. I am a patient advocate. My daughter has a genetic disorder. It's not an SMA, another one under congenital muscular dystrophies. But of course, even though I am an engineer, as you can imagine, we have happened to be kind of a gene engineer actually. Anyway, so thank you very much, Milos, for this presentation. Over the past 5-6 years, this is my understanding. Please correct me if I'm mistaken. But over the past 5-6 years, I have been seeing that there is kind of sort of an era opening actually in technology in terms of these gene therapies, etc. And SMA is one of the leading ones, of course. So I have been trying to understand. We have been in touch with a great team from NIH in the US, not only for my daughter, of course, for the other kids having these serious diseases. So I would appreciate from you and from all other scientists and MDs getting involved in these diseases to show the progresses on on the kids who have been receiving these gene therapies and disease, because it's not easy to convince people, especially who are getting involved in fundraising things, because they don't believe that they still, even some MDs still think that they are under clinical trials. They don't know that they have been approved. So people should understand and see and be convinced that these diseases, these drugs have been helping these kids a lot. So I would appreciate to share more videos or other pictures showing that these drugs are working pretty well, because I personally know, but of course it's not easy. So that's gonna be, that would have been my comment. Thank you very much again. By the way, thank you very much for this conference. I just chatted with Arabella in between. So thank you very much who has contributed to this event. Thanks a lot again.
Thank you for the comment. I think it's very important to keep talking about these things, because I really believe that people are very afraid of these new things. Like when people in Serbia hear that we are going to perform the genetic newborn screening for SMA, that we are going to extract DNA from a dried blood spot, that's the moment when they try to refuse the screening, because they think that they will that we will do I don't know what, like clone their child or do some experiments on their DNA. So whenever people hear something regarding DNA or RNA, they have this like inborn fear that something wrong is going to happen. Maybe the reason is the COVID-19. At least in Serbia, we had a very bad campaign regarding vaccines. So I think that people completely lost their faith in science and medicine. And maybe this is just like the time that we are living in, like the conspiration theories, or I don't know. So I'm very thankful for the comment that you made, because it's extremely important to keep talking about these things, to show their efficacy, but also to keep talking in a very simple way, so that somebody who is not from the field can easily understand the benefits. Because what I'm trying to at least highlight here when I'm talking with people is that everything that we are doing in the newborn screening for SMA is in the best interest of the child.
Thank you very much. I've seen a raised hand.
Yes, hi. Yes, I actually, the question is a follow-up from what you said. So you said at the beginning that the uptake was very poor of the screening, and then it improved over time. So I guess lessons learned for other countries that don't do that yet. Did you do something? What happened? How did you persuade the families
actually,
that other countries can follow?
We put a lot of media pressure on the country and on the project. So in a short period of time, we became like little stars here, because we were appearing constantly, daily on the TV, talking about the project. Because the point was to let people know that it's a good thing. People were afraid, so we didn't want to like draw any negative conclusions from these rejections. We knew that people were afraid, so I think that we have to post things about this on social media, on Instagram, on Facebook. I was writing articles, sharing them with the people. I was talking publicly on TV. I was talking about this whenever I could, wherever I was. So I think this was our way to reduce the number of rejections. So in the beginning, it was not that poor. We had around maybe 5% of rejections, but now almost every family accepts the screening, because and of course, as I said, this is not only our success. The country was already afraid of SMA. People were afraid because they heard about these numerous fundraising campaigns. They heard about the children that were affected by the disease. They saw videos of these children. They saw pictures of them. They saw how severe the disease is. And that's all together, it raised the awareness and reached the target population, stimulated the enrollment into the project.
Thank you.
Thank you very much. Thank you very much. This is a great lesson to learn for Eastern European countries to follow your stepwise approach. And thank you for sharing this with us. And even if we wouldn't believe, we are just in time for lunch now. And I wish you to have a good lunch. And we see each other in 1 hour. So thank you very much, all of you, for your involvement and for your participation. See you later.
See you. Goodbye.
Bye.
Tubes for feeding. The second drug that is approved in few countries around the world is Adravon. It has an antioxidant agent action and was first used to treat stroke patients. It's not registered by European Medicines Agency, but it just slows the rate of functional loss by over 33%. There is no data on the survival. There was a very recent German study completed, which didn't find any difference between edaravone and riluzole against riluzole only. It was available for venous treatment, but since this year there is also oral pills available, which is by far most convenient for patients. These are the only drugs that are registered. There's also two other drugs, Tutka, which is Tauro Urso, ursodiol and phenylbutyrate. This is two substances, two molecules that together prolonged survival and reduces the number of hospitalizations. It is only second stage of the drug trial that was finished, and sodium phenylbutyrate and tauroursodiol, they are kind of affect cell function. There is very little of potential side effects. These two drugs are since September this year registered in USA as Librio and in Canada as Albrioza. We are happy here in Slovenia that are using these two drugs since June last year already. There is many ongoing clinical trials that are listed here that are still not finished yet. And this is mainly, I'll switch now to the gene and stem cells therapy. There is several possibilities to use gene therapy. One is use of antisense oligonucleotides, and the other is therapy based on viral vectors. The trial has been finished. It is on tofersen that is used to treat patients with SOD1 mutations, while stem cells, they are actually targeted to normalize cellular microenvironment and not to replace motor neurons. There is no definite evidence yet as to their safety and efficacy. They might cause tumors, for example. As to the conclusions, the last 3 decades have been have seen much hope and as well much disappointment for promising pharmacological therapies. Only 2 drugs, riluzole and edaravone, have shown slightly positively effects on the course of the disease. This is very likely the result that the disease is very heterogeneous with regard to the clinical presentation and pathogenesis, and it is very likely also that a single therapeutic approach is likely to be ineffective for all patients. And there is much hope because of the deciphering of many pathogenic mechanisms, especially in these genetically determined disease patients. Thank you very much.
Thank you very much. And now I'd like to invite Stanislava Ristikova-Jek to present her speech about ALS group at the Institute of Clinical Neuropsychology. Thank you very much, and the floor is yours.
Yes. And screen. Open. Sorry.
We can see the slide.
Yes. You see? You.
It's okay.
Okay. Okay. Thank you for word. First of all, I'm grateful to Mrs. Andrea, the biggest advocate for ALS patients by the Society of Dystrophy of Slovenia, for the invitation that today we can present the history of ALS team in Slovenia. While last month, ALS team was approaching 20 years of organization. Thank you, Andrea, for introducing me to the audience. The idea to organize integrated care for patients with ALS has begun in 1997. At that time, I have been familiar and alert with ALS by neurologist Dr. Janez Zigar, who has treated people with neuromuscular disease. The idea has been immediately put into the practice and in 1997 we started actually tried to treat the patients with ALS holistically. A person with his own medical history, desires, values, family situation, social circumstances and lifestyle, what play into his experience of health condition or disease. Firstly, we started the discussion to ourselves. I remember that at the time I was in the role of nurse, of dietitian, speech therapist, psychologist, and etc. Depending of the course of progression, ALS disease can be a parable, as for vortex, as for spiral. Usually one problem is followed by the second and fourth, and to take quick action is required. Patients and their relatives usually find it difficult to adapt to rapid deterioration. Quick action means the organization of established routes of medical care and assistance in order to be able to at least partially alleviate the patient's problems and concerns. What is relevant today may not be necessary in a few weeks or months. For example, electric wheelchair. In October 2002, it has been formed organized patient care with a group of professional colleagues known as the ALS team. At the same time, within the framework of the Society of Dystrophy of Slovenia, patients with ALS have organized themselves into the ALS committee. At that time, there were 2 neurologists, a speech therapist, a hospic, a pulmonologist, a gastroenterologist, representatives of the ALS committee, and myself as a social worker and coordinator. Over the years, more and more professional colleagues have joined us. At the beginning of the organized system, we set about creating protocols. Nowadays, the ALS team covers outpatient hospital activity. The patient is usually referred to a neurologist by his personal physician, usually followed by a diagnostic procedure. The next step is communicating the bad news or talking about the illness. It's usually a family gathering. We offer patients the options of further treatment in our team of ALS. Usually, checkups, frequency or re-examinations are on average on every 3 months. During the comprehensive treatment, we also cooperate interdisciplinary with experts from outside of our institution. We advocate a palliative approach to a holistic treatment. As the disease progresses, we also take care of feeding and breathing problems. If necessary, in these cases, patients are also admitted to the hospital treatment. Patients and their relatives can always contact the coordinator of the ALS team. The team approach enables timely identification of problems and quick action. At the same time, better care reduces the number of hospitalization and improves the quality of life. In 20 years, we treated over 740 patients, of which approximately 390 were men and almost 350 were women. So the number of new patients increased yearly by about 30 in recent years. The average years is 65 years. So a review of the availability and organization of ALS medicines. In 1998, 5 patients have been allowed to participate in research with the medicine Riluzole. 7 years later, in 2005, Slovenian patients have finally got the rights to the medicine Riluzole. At that time, it was called Rilutec. Later on, there was the competition with Sklefic, and in 2020, the possibility of the medicine in liquid form Teglutic, which made it easier to care for patients with swallowing disorders. In 2021, our health insurance company supports our request for a new medicine against ALS disease, namely phenylbutyrat, as Hemonaps or Ravicti, which patients today can get after applying for it.
Sorry to interrupt you, Dr. Stanislava. Can you maybe try to press F5? Maybe we can then see the whole screen. Our IT guy said that if you try to press F5 from your keyboard, it may work.
This? Sorry.
Yeah, no problem.
Sorry. Thank you. Thank you. Yes, thank you. For the treatment of spasticity against spasms and against excessive salivation in the pharmacy of the University Medical Center of Ljubljana, we have provided, arranged the availability, the medicines that are not covered by the health insurance company. So we have also arranged accessibility to some customized medical devices and interventions. At the beginning of our treatment, we were able to help patients with breathing disorders with CPAP devices. And in 2006, we have started using BiPAP devices for breathing and later on even respirators. Sorry. We agreed with the Rehabilitation Center for customized neck braces for patients with communication difficulties. The insurance company arranged for the rental of a replacement communication device. We have also made sure that patients get priority to access customized peroneal braces. In 2003, we have asked for a wheelchair to be rented. In the same year, we also took care of the speaking program, which facilitates communication for patients who were left speechless. In 2007, we have asked the Society of Dystrophies for a co-suppressant to lend it to the patients. In the case of swallowing difficulties, we solved the problem with the classic insertion method of PAG. Nowadays, the feeding tubes are designed with more modern, easier procedures for the patients. It is also a novelty that our mobile ALS team can change the feeding tube if necessary, that the patient doesn't have to go to the hospital. So other activities of Team ALS, in 2004, we have published a leaflet about the disease. In the same year, the clinical psychologist has organized a monthly meeting for caregivers and relatives of ALS patients. We have connected to the Golnik Hospital, which organizes education for relatives in case of decision for invasive ventilation. Yearly, we send notification to patients about vaccination against influenza, pneumonia, and against SARS-CoV-2. We wrote the criteria for admitting patients to the hospice house. We wrote we are involved in the Butterfly project, which follows a palliative treatment approach. In 2020, the health insurance company has approved home visits for those patients who are in the last stage of the disease and an artificial ventilation. At the beginning of its activity, the Committee for ALS published a newsletter Lastovka Follow, where we are participate with our contributions. We were also guests on Radio Slovenia, and the show about ALS was hosted by a journalist who also has diagnosed ALS. If you remember, worldwide, known Ice Bucket Challenge in 2030, we also doused ourselves with ice cold water in Slovenia. At the Slovenian Book Fair in Ljubljana in 2006, Mrs. Andrea and me were guests at the release of an autobiographical book written by a Swedish journalist who lived with ALS disease. In the same year, we were guests at the publication of a book in the form of diary entries of an ALS patient who had already died. In 2021, we participate to the first manual on palliative care in Slovenia, which was published. Otherwise, our colleagues are very active in researching and writing scientific articles related to the ALS disease. For better recognition of the disease, we also organize lectures in retirement homes, general hospitals, and colleges. In 2017, the European ALS Association has organized a meeting with a symposium in Slovenia. Professional colleagues in the team also carry out many researches where patients are involved. Professional colleagues are also receive additional training on regular basis. So, as part of the ALS team, we had also managed to organize 2 one-day trips. At the end, there are wishes of our ALS team. There are certainly many of them, but I would like to highlight 3 mains. Increase the database, which is currently only medical. The second is update the website, and the third is telemedicine. This is the conclusion of my presentation. Thank you for attention.
Thank you very much for this presentation on integrated care for ALS patients and for your experience over 20 years in caring these patients. I would like to open now the floor for questions and comments or do you have any questions? I'm looking for raised hands. If you can unmute your microphone and ask questions. If not, I would have a question. How difficult is to ensure coordinated care for these complex conditions like ALS? How difficult it is to organize this care and to bring together all the professionals to bridge the gaps between social, medical, and educational services, because you are talking about social services, medical services, but also trainings and so on. How difficult it is to organize this?
This is difficult. Yes, I don't know how to organize, but I have good colleagues who is I don't know.
Do you have some case managers, for instance, people that help people?
No.
Okay.
No managers. I and Dr. Zidar is main in this team, and colleagues work with us. I is non-stop, is always in the ambulance with neurologists, and this is the catch how I can make this work.
Yeah. Okay. Do you have any questions?
Dorica, there is a question in the chat from Dr. Raul. He's asking if is a radiological intervention of any help to ALS patients. So if you use radiological intervention.
Yes, we use, but radiological intervention work, radiological doctor for example, which when we need to make a PAG.
Okay, thank you. Thank you very much. Are there any other questions? Somebody is asking you if, are you aware of any cured cases?
Yes.
Okay. There are no other questions. I would invite our next speaker, which is Ioana Streața, Dr. Ioana Streața from Romania. She is a medical geneticist at the Regional Center for Genetics in Craiova, and one of the main active experts in our national network for congenital anomalies with or without intellectual disabilities, which is member of ERN ITACA. She will speak about genetic testing in neuromuscular diseases, because understanding testing methods, interpreting complex results, and dealing with the ethical, social, and personal issues that arise for patients and family is critical for their care. Ioana, we are inviting you to tell us more about this topic, and welcome to the conference. Thank you very much for being today with us.
Thank you very much, Dorica, for your nice presentation. Thank you very much, Arabella, for the invitation to join you today here. And I will start my presentation. Just a second. So I'm allowed to share my screen?
Yes, you should.
Okay. So now can you see my presentation?
Yes, we see.
Perfect. Because now I will put it on a slideshow, and it will be better. So thank you very much again for the opportunity to be with you here today. And during the next few minutes, I would like to make together with you a journey in the complexity of genetic testing in neuromuscular diseases. The topics that I want to talk about today are a brief review of neuromuscular diseases, the link between genetics and neuromuscular diseases, and how we can use genetic testing in the diagnostic of neuromuscular diseases, taking here looking here to few details like genetic methods, genetic testing algorithm, genetic counseling, and familial genetic testing. And I will outline the importance of multidisciplinarity in neuromuscular diseases, and just to show you all these ideas in practice. I will present a case report with a neuromuscular disease that was managed in our center. So, neuromuscular diseases are disorders of muscles, neuromuscular junction, nerves, and neurons from the central nervous system, and that's why the clinical presentation of these pathologies is very heterogeneous. It's complicated. Usually, it's very complicated to give the proper clinical diagnostic and to put the proper name of the disease. The majority of these neuromuscular disorders have a genetic cause, a genetic background, and these genetic causes are most of them inherited or transmitted in a Mendelian way. So they are caused by mutations in genes that can be transmitted in a dominant or a recessive way. I will outline this a little bit later in my presentation. So, because most of these pathologies have a genetic background, a genetic etiology, genetics plays an important role in, or might play an important role in the diagnostic and management, but is not the only tool that we have to use in the diagnostic and management of this kind of pathologies. As I've mentioned, what is so we are, as I like to say, we are a library with 46 volumes, with 46 books, and these 46 books are arranged in pairs. Each pair each book pair has two variants, a maternal one and a paternal one. And in all these books, we have stories that we name in a scientific way genes. These genes are can be sometimes can contain errors, typing errors. The letters from them can be substituted, deleted, or duplicated, and these are named or known as gene variants. These gene variants can be inherited or they can occur de novo. In all this library, in all these 46 books, we have around 30,000 stories or genes. And from these stories, at this moment, we know that around 500 of them are involved or are causing neuromuscular diseases. The number is changing very, very fast because the progress of technology and of science help in this direction. Medical genetics, because of this, is a very important tool. Medical genetics is a medical specialty that can offer clinical help and clinical diagnostics, but in the same time can confirm through laboratory tools the clinical diagnostic. And it has two components or two arms in this specialty, but all these phases of medical genetics have as main target or main activity not only the patient, the individual, but also his or her family, and in this way the entire population, because a genetic disease is not only the disease of the individual, is not affecting only the individual, it affects the family and also the people around. So it's important to have and to find the genetic cause of the pathology. As I've mentioned, the variants that can be found in the genes in our stories can be inherited or can be de novo. And if they are inherited, they can be inherited in a dominant way or in a recessive way. If we talk about the books that, or the chromosomes that are, let's say, we call them autosomes. So, but they are, if we refer to the books that are involved, are responsible for saying the gender of the person, X and Y chromosomes, then we talk about an X-linked recessive, an X-linked inheritance that can be recessive or dominant, or we can talk about an Y linked inheritance. And also, we have an extra book in our library, and this is not this book is called the mitochondrial DNA, and this book can be also contains also stories, genes that can be also involved in or can be involved in developing or causing neuromuscular disease. So it's important to know all of these data and details, because that's why there is no perfect genetic test that can offer us from the first moment, from the first time, the diagnostic. Yes, maybe we can say, no, this is not true, because now we have whole genome sequencing that can offer us in the same time the analyze the assessment of a point mutation with copy number variation, with so small errors, with big errors, or and in the same time the mitochondrial DNA. Yes, it's true. This is one of the most complex genetic tool that we have now, but sometimes there is something else that can cause this pathology, and we can talk about epigenetics, but this is not what I'm going to develop more, because it's complicated. So this is the way that the genetic variants can be inherited or can be transmitted from one generation to another. That's why through genetics, through medical genetics, we can help to prevent, to have an early diagnostic and proper management for genetic diseases. And we can help not only the person that is tested at the beginning, but we can also help the family and the population from the region from where the patient is. And now, having in mind this library with the books, with the stories, and with the letters that are used to write these stories, I will present you a little bit about the genetic tests that can be used for the diagnostic of neuromuscular diseases. First of all, why to make a genetic test? Because we want to confirm a diagnostic of that was clinically suspected based on the phenotype that the patient or the individual presented. But in the same time, we want to offer a personalized and management of the individual and his or her family in order to achieve a high quality life. We can also, through genetic tests, avoid invasive or painful procedures such as muscle biopsy, but not always. This might be possible because sometimes after negative or uncertain result, the genetic test, but with a very let's say specific phenotype of the case, muscle biopsy is needed and can help. And in the same time, we can give the opportunity through genetic test to the patient and his family to make family planning, but also in the same time to participate in research studies dedicated for that specific condition. Also, disease management can be helped because we can try to provide prognosis for the severity and the progression of the disease, and we can guide sometimes the treatment and the management based on the genetic test result. As I've mentioned, because the genetic causes that can produce a neuromuscular disease can be from the can be some of them very big, like dimension, but some of them very small. So if we look to the if we use again the idea of the books, sometimes chapters might be missing or might be extra in a book, but sometimes in a single story, in a specific in specific coordinates, one single letter can be different, can be changed, can be substituted by another letter, or can be deleted or can be duplicated. And this is a very small genetic variant that requires, let's say, specific genetic methods to be identified. So there are a lot of methods, genetic methods that were developed and can be used in the diagnostics. So the first genetic methods were the cytogenetic methods, the karyotype. Then the karyotype was improved and it was developed the molecular karyotype that can detect the first one, the classical karyotype can analyze the books, the library, the entire library can count the books and can say that there are missing or are extra big parts of the books there. After, the molecular karyotype can count chapters in each books and can say extra that there is a chapter that is missing, or there is a chapter that is duplicated. But if we want to read the books, we can, or to read a specific story from this book, we need other type of methods, of genetic methods. And these are the methods based on sequencing. And if we want to read a single book, a single story, or we want to look for a single variant in a specific story, we can use capillary sequencing methods because they are, let's say, cheaper and faster. But if we want to read all the books from our library, to read huge stories, huge genes, and we do not know where to look first, because there might be many genes that can be involved in the phenotype of the patient, then next generation sequencing methods are those that can be used and should be used. Because if we use other type of method, then the result will be negative, but not because it's nothing there, just because we didn't choose the proper method. If we look to the NGS, to the next generation sequencing methodology, that is one of the most complex and strong and powerful method, we can choose to analyze a specific panel of genes that are very well, that are strong associated, very strong associated with neuromuscular diseases, or we can choose to analyze the whole exome or the whole genome if we want to be sure that we analyze all the genes and all the regions of our genome that might be associated with this kind of pathologies. And of course, if we talk about whole genome sequencing, we can include also the analysis of mitochondrial DNA that can also be involved in the development of neuromuscular pathologies. The genetic testing algorithm is complicated because of the things that I've mentioned before. There are a lot of questions that we have to answer before we choose and we say, this is the genetic test that you should do, or I recommend to you. It's important to know why we test, what we test, who we test. Is the person is affected or has the history, has a familial history for a disease, a neuromuscular disease. When we want to do this test, prenatally, postnatally, we want to make a screening, and how. When we have the first time when neuromuscular pathology might be suspected, usually is in childhood, and the first specialists that can raise this suspicion are the child neurologist or the pediatrician, and they and it is recommended to have, for those cases where is this kind of suspicion, to have a multidisciplinary assessment before genetic testing. Why? Because it's important to decide which test to do. Maybe we can say yes, we can recommend to all whole exome or whole genome sequencing, but it is about costs, but also about limitation of the methods, because sometimes even these two methods can give negative results or can have can't give the response of which genetic variant is involved in this phenotype. So it's important to discuss in a multidisciplinary team, and only after this to decide the genetic test that will be recommended. And also, if we want to perform a whole genome or whole exome, ideally is to test the trio, the parents and the child, because it will be easier to find the variant that might cause the phenotype. And of course, we have to choose and to say if it's targeted or it's a screening, let's say a genomic screening method. As I mentioned, also it's important when we test prenatally, postnatally, screening, or preconceptional, because in each of these parts, it's important to know what we test and which is the proper method we want to use. Also, after we've done a genetic test, sometimes it might be necessary to repeat that test. And of course, we repeat the test if we performed just a targeted test, or we performed a panel gene test and not a whole exome or a whole genome. Because if we performed whole exome or whole genome, the approach is not to retest, the approach is to reanalyze the data in the context of new data in the literature and in the databases. Also, sometimes genetic testing can be done through research studies, and it's, let's say, just research use only the result. And if we want to have access to different to new therapies that are on the market, or we need sometimes, or to have the test done in a diagnostic laboratory. That's why it's necessary in these situations to repeat the test. And of course, in some clinical trials, the methodology of these ask for a new test to be done in specific laboratories, because these are the rules. Before, after, and whenever is necessary, genetic counseling is recommended, because during genetic counseling, individuals and their families can be informed about the limitations of a genetic test, the variants of genetic testing that are available, how can help the genetic test, and which should be the expectations we should have from the result of genetic test, because it's important to know that in many cases, the genetic test will tell us only this is the cause, this from here everything started. And of course it's important because we can look and find arguments in the literature about a proper case management, but won't offer us a treatment option. And it's important to understand that and to accept this limitation for many of the situations. And after, and it's important to know, and genetic counseling will help us to understand that after we have a genetic result, we can test also members of our families if they want and they need the genetic test. And we can also, and it can be also necessary to test our parents, for example, or our siblings, if we are the first person in our family that was tested and where a genetic variant was found, because it's important to establish the inheritance mode of that variant. But always genetic testing is a personal decision. The genetic counselor or the geneticist just recommend, explain, but always the patient and his or her family will decide if they want to do or not the test. As I've mentioned, or what I wanted to outline through my slides, is that genetics and medical genetics recommends a multidisciplinary approach in the management of neuromuscular diseases, because the genetic diagnostic is just a small piece from the puzzle of neuromuscular diseases management. And the genetic test is just a strong tool that has to be used carefully and wisely if we want to obtain more advantages than disadvantages. And as I've mentioned, I will conclude with one case report from the regional genetic center from Craiova, Romania. Is the case of a patient with clinically diagnosed with Charcot-Marie-Tooth. She came to genetics because she wanted to have the genetic confirmation of her disease, and because of preconception genetic counseling. So the first step, after we also see clinically, clinically the case, was to try to identify the genetic cause. And at that moment, because one of the most frequent cause of Charcot-Marie-Tooth is a duplication in one gene, in PMP22, we tried to identify, to see if we had identified this copy number variation, duplication of this gene through RACGH, so through molecular karyotype, and this result was negative. After that, the patient was able to continue with the genetic testing abroad, and she performed panel sequencing, gene panel sequencing of the most frequent genes that are involved in Charcot-Marie-Tooth. Also, this one was negative. A few years later, when we were able to implement and in our laboratory the next generation sequencing method, we were able to offer her genetic testing of a clinical exome. We named a bigger panel, gene panel, with more than 4,000 genes. And this is also known as clinical exome. It's not the entire exome, but are all the genes that are associated with specific pathologies. And this time we find there a variant, a genetic variant in a gene called NDRG1 gene that is associated with Charcot-Marie-Tooth type 4D. And when we compare the clinical phenotype of our patient with the clinical phenotype described in the literature for this Charcot-Marie-Tooth type and the gene, we could say that there is a correlation between the genotype and the phenotype of our patient, and that is the genetic variant that caused the problem to our patient. After this, we were able to offer her the result. We were able to counsel her and say which are the next steps that we and she and her family have to do, because there were several steps to perform. We validate first the variant found through this method using a targeted method this time, capillary sequencing. Then we continue to test the family, the mother, the siblings, and the husband, because this gene is, or the type of Charcot-Marie-Tooth 4D is transmitted, or it's autosomal recessive disease. So requires two errors, one on the maternal book and one on the paternal book or story, to cause the disease. And our patient was homozygote for the mutation, but it's very the probability that her husband to be carrier of that variant, it's, let's say, very low, but you never know, so you have to test this, and that's why we tested also the husband of our patient, and of course he was negative. We tested the family, the mother, and her sister and her brother, and all of them were negative, so wild type homozygote. They have no variant there. And of course she was we tell her that there won't be a problem if she obtains a pregnancy, but just to be sure, it might be better to test prenatally the fetal DNA in order to see that he or she carries only one copy of the variant and no other de novo variant occurred there. And this is what we've done. We've done also prenatal diagnostic, and of course the baby was heterozygote, so healthy carrier, and everything was with happy end, because she gave birth to a very, very healthy boy, and very beautiful. And we were able to solve a case, to solve not only a case, to help a family to be happy and complete. So we excluded the CMT4D in other members and in the fetus, and then the child, the unborn child, but he was born, and everything went okay because the patient, our patient was patient, and we were able to find all the tools that we needed to help her. But of course, there was some big time to wait between the tests. Our message is never alone. We have to work together, doctors and patients, in order to have an integrated multidisciplinary management of each cases. Thank you very much on behalf of the team of the Regional Genetic Center from Craiova. And if there are questions, here I am.
Thank you, Ioana, very much for this overview of genetic testing and the example in the end with happy end. And I'd like to open the floor for questions and comments. If not, it means that everything was clear. Sorry?
Yeah. Sorry,
Okay.
Dorica. No, just a comment. Very nice talk. Thank you very much. I just saw that in the process, at the end of your process, you were saying that once the genetic mutation has been identified, it's important to report this data in a registry. So I just wanted to share an information with everyone in this meeting. More and more, there are standards which are promoted for registries following a few principles called FAIR principle for findable, accessible, interoperable, and reusable data between registries. And so I had two information for you. First, the ERN Euro NMD, which will be presented tomorrow, I think, is building a hub where existing registries can connect and share their data. So that's very important for all the clinicians who are currently managing and building registries to know that it's still in the building process, but in a few months or years, hopefully months, they will be able to connect their registry to this hub so that the data can be shared at the European level as much as possible. The second thing is that when we talk about this FAIR data, that means also adopting certain standards so that they are interoperable between different registries. So tomorrow again, there will be a presentation of the registries by Nawal Lalout from WDO, or Duchenne Data Platform, sorry. She will show how they adapted the Duchenne Data Platform registry to make it fair. But more importantly, she will tell that the tools they developed has been delivered as an open source software. So it can be adapted to any existing registry. So if you have an existing registry and want to connect your registry and make it fair and interoperable with other registry in the world, you don't have to restart your registry from scratch, but you can use this open source software to put it on top, on additional layer on your registry, and then have it connectable to any other existing registry. So yeah, I think this genetic data are very important as we are talking about rare diseases. And to make sure that we clearly interpret these mutations, we need to be able to compare them with a lot of other patients sharing the same symptoms. And so again, data sharing is a very important thing.
Thank you very much. Just a question or maybe need of clarifications. The registry that you are mentioning is developed within the neuromuscular disease European reference network, no?
So yeah, I talked about two registries. So one is really the hub developed by the ERN, so the European Reference Network EuroNMD. And as its name mentions, it's more a hub where existing networks, existing registries will be able to connect their data. And the second registry is the Duchenne data platform, which is a Duchenne dedicated registry, which had to be adapted to be connectable to the ERN hub. And to do so, they developed these open source tools, which allow any registry to be easily connectable to other registries.
Okay, thank you.
Thank you very much, Ioana. Thank you very much, Alexander, for this useful comments and information for everybody. And we are going to the next presentation. Caring for patients with neuromuscular disease is challenging, and the respiratory care is of the utmost importance because it is a major determinant of quality of life and survival. Professor Sedat Oktem is the professor of pediatrics and head of Department of Pediatric Pulmonology, Medipol University Faculty of Medicine in Istanbul, Turkey. He is member of European Respiratory Society and member of the Turkish Thoracic Society Pediatric Chest Diseases Working Group Executive Committee. His main research interests are the home ventilation, tracheostomy, care, aerodigestive disorders, rehabilitation of ventilator-dependent children, invasive and non-invasive ventilation. Also, Yasmin Gokdemir has been working as a professor of pediatric pulmonology in Marmara University in Istanbul. Her interest areas are cystic fibrosis, pediatric lung transplantation, neuromuscular disease, and sleep disorders, breathing in neuromuscular diseases. She had been in pediatric lung transplant program in Pittsburgh University and Boston Children's Hospital in 2015. She is currently head of Turkish Thoracic Society Pediatric Pulmonology Assembly. We are inviting now Professor Sedat Oktem and Professor Yasmin Gokdemir from Istanbul Medipol Universities to bring more information with the presentation respiratory in neuromuscular patients. Thank you very much for being today with us.
Thank you. Now I will share my presentation. Can you see my presentation in full screen? Okay, good. First, I would like to thank the organization committee for their kind invitation. In Marmara University, we follow up almost 200 children with neuromuscular disease in close collaboration with pediatric neurology and physical therapy and rehabilitation team. And almost half of our patients are SMA patients. Because our center is one of the SMA centers in Turkey. In Turkey, nusinersen treatment is reimbursed. It is the only reimbursed treatment option for SMA patients. So 88 of our patients are under nusinersen treatment. And I am happy to announce that SMA newborn screening has started in Turkey in this year, and 4 of our patients were diagnosed by newborn screening. And here you see the cover of the first book in Turkish about the respiratory problems of children with neuromuscular disease. And this book is published by Turkish Respiratory Society, and I am happy to be one of the editors of this book. And in Turkey, as Turkish Pediatric Pulmonology Society, we perform hands-on courses and workshops to increase the knowledge of the physicians and healthcare providers. And Professor Oktem will mention about this later. Now I move to the main topic of my presentation. And neuromuscular diseases causes decreased upper airway tone, and chest wall incompliance, and weakness of the inspiratory muscles, and also dysphagia. And decreased upper airway tone leads to obstructive sleep apnea, and then impaired chest wall compliance leads to chest wall deformity and scoliosis, and weakness of inspiratory muscles leads to inadequate airway clearance and recurrent respiratory tract infections. And also dysphagia and swallowing problems cause pulmonary aspiration and recurrent respiratory tract infections, and unfortunately bronchiectasis and fibrosis. And all these underlying abnormalities cause chronic respiratory failure in children. So the most common cause of unplanned hospitalization is acute respiratory failure associated with respiratory tract infections. Here you see chest X-ray of a patient. On the left side, left lung, it was obstructed with thick secretions, and picture at the bottom shows the thick mucus that's seen during the bronchoscopy. And after the prompt treatment of infection and also intense airway clearance, you see that the left lung has opened and children's respiratory distress removed. So this is very important that the patient with neuromuscular disease should also be followed up from respiratory therapy from the pediatric pulmonologist. And unfortunately, the most common cause of death is chronic respiratory failure in these children. But neuromuscular disease is a huge umbrella, and age of the onset of respiratory problems depends on the disease type. So every patient that diagnosed with neuromuscular disease should also be referred to pediatric pulmonologist. And what we do during our outpatient clinics, we do clinical evaluation of the patients, evaluate the respiratory functions, evaluate the strength of respiratory muscles, and also cough peak flow rate measurement is also very important. We do annually blood gas analysis, and monitoring the nighttime sleep is also very important, and evaluation of the aspiration is also very important in these patients. And how do we do clinical evaluation? And we assess if the patient has any change in voice or cough, difficulty in clearing secretion, in feeding and chewing, and we examine the patient's posture, if the patient has kyphosis or scoliosis, and we evaluate the growth and also nutritional status, and if the patient has increased frequency and severity of respiratory tract infections. And we should also assess for sleep disordered breathing in every examination. And we do spirometry to measure lung function in children that are capable of forced expiratory measurements. Here you see a patient doing spirometry sitting and in lying position. And we do tests to assess respiratory muscle strengths. And these tests usually correlate with spirometry, and these tests are easier than the spirometry. Here you see a patient during the measurement of inspiratory and expiratory muscle strength, but these tests also need cooperation. And here you see a patient, this is our patient diagnosed with Duchenne muscular dystrophy. So these patients can perform these tests easily. And we here measure the inspiratory muscle strength from nose of the patient. And cough peak flow measurement is the most important measurement type of the respiratory status, the cough efficacy, and if the patients can effectively clear their airway. This is a very important and easy technique to understand the patient's respiratory status. And we do annual blood gas measurements in patients to diagnose the sleep disordered breathings. And another important evaluation is the sleep monitoring. we can do sleep monitoring with different methods. Pulse oximeter is an easy method, but gives not so much information. Oxycapnography is much better than measurement of only pulse oximeter, but the major and golden standard method is polysomnography and overnight polysomnography. But this is a complex laboratory analysis. And here you see a patient from our lab. He doesn't seem so happy with a lot of cables, but we usually don't face many problems with these sleep studies. But you need to have a very well-trained technician in your sleep lab. And evaluation of aspiration is also very important because difficulties in swallowing increase the risk of aspiration. And don't forget that recurrent pneumonia may occur due to silent aspiration. So if the child or the caregivers don't give and don't say about aspiration, you should still evaluate for aspiration in children with recurrent pneumonia. And now I will move to the treatment of respiratory problems in neuromuscular disease briefly, because Professor Öktem will go in detail with this. Clearing the airways are very important, and there are many methods. And the caregiver and also the physiotherapist choose the best way for the best method of airway clearance for the child. And when the child gets older, the method should be evaluated again and maybe switch to another method for this patient. And also cough assistance is also very important. It can be done manually or with some devices. These devices are not reimbursed in Turkey, so many of my patients don't have these patients, so we use manual techniques for cough assistance. And in non-invasive ventilation is also used to treat the nocturnal sleep problems and to reduce the frequency of hospitalizations due to respiratory tract infections, to prevent the development of chest wall deformity, especially in SMA patients, and it helps to prolong the lifespan. So it's a very important method for respiratory support in children with neuromuscular disease. And there are a lot of masks that were invented for children. So if this is an I recommend the caregivers don't afraid of starting these devices, because they will help your children so much. And tracheostomy is the last option, of course, tracheostomy and home ventilation. But patients who cannot be extubated more than 2 weeks, or those whose hypoxemia or hypercapnia cannot be treated with non-invasive ventilation with BiPAP devices, or some may have severe mid-facial hypoplasia, then tracheostomy is a chance of the treatment. And treatment of accompanying problems is also very important, such as gastroesophageal reflux. And nutrition with gastrostomy is important to maintain the nutritional status in these children. And to prevent infections, vaccines are very important, especially pneumococcal vaccine, annual influenza vaccines, and also prophylaxis of RSV. This is my last slide. As a result, lung problems are important causes of morbidity and mortality in neuromuscular disease. It is important to monitor respiratory findings. Clinical signs should be questioned at every examination. Spirometry should be performed if possible, and cough peak flow meter measurement is also very important. Blood gas analysis and nighttime sleep monitoring are recommended once a year, and non-invasive ventilation with BiPAP device occupies an important place in treatment, and vaccines, infection control measurements, and protection from cigarette smoke exposure are important in follow-up. Thank you for your attention, and I will be happy to answer if there is any question.
Thank you. Thank you very much. And we are open for questions and comments. Okay, if we don't have any questions, we are going to have our next speaker, which is bringing a question mark. Can artificial intelligence in MRI help us understand
Dorika, sorry, we passed Dr. Sadat because they were supposed to present together. So we still have, sorry about that.
Sorry, I didn't understood how it is going to be done. So sorry, Dr. Sadat. So I already introduced to you, so the floor is yours. Thank you very much.
Good afternoon for all. I am happy to join with you. I am presentation about what to in Turkey. I have no conflict of interest, and also I have permission to use about my pictures during presentation. As you know, management of neuromuscular disease is a key team playing. Multidisciplinary approach is the key element of the management of neuromuscular disease. A lot of members consist of this team. For example, pediatric pulmonologist, gastroenterologist, dietitian, language therapist or swallowing therapist, orthopedist consist of team. For this reason, we want to create a team in Turkey. At the beginning, 5 years before, there is no home ventilation program in Turkey, and there is no standard tracheostomy care guideline, and there are few healthcare providers or doctors can be performed non-invasive ventilation, mechanic insufflator, and high-frequency chest wall oscillation device. And also families and caregivers have little information about tracheostomy care, tracheostomy changing, NG tube replacement, airway clearing device, and ventilator troubleshooting. Also, there are a few centers can be performed non-invasive ventilation, sleep study, and swallowing evaluation, such as flexible endoscopic swallowing study or video fluoroscopic studies. For this reason, we collaborate with Turkish Pediatric Pulmonology Association, and we prepared some pictures and videos for tracheostomy care. For example, aspiration, tracheostomy changing, skin care, accidental declination, etc. After that, we prepared tracheostomy booklet for healthcare providers and caregivers using these pictures and videos. Also, our patients can be reached freely in this association website, videos and booklets. After that, we prepared some training slides or training sets for tracheostomy care for healthcare providers. 2 years ago, we held training program for tracheostomy care in the simulation room. This trained healthcare providers can make the hospital and their patients educated or trained by this. Sorry, I can't see. These healthcare providers can make their hospital and they trained their families and caregivers for tracheostomy care and changing. After that, we focused on ventilatory troubleshooting because there are a lot of problems, patients suffering ventilatory troubleshooting problems. And we will planning 1 year later for the ventilator troubleshooting program for patients. Also, we organized some session in Turkish Thoracic Society and also Turkish Pediatric Pulmonology Association Congress for the neuromuscular disease ventilatory support, about the ventilatory support. Also, we prepared some book for healthcare provider for the non-invasive ventilation and the neuromuscular disorders respiratory care. Also, we collaborate with other colleagues, for example, pediatric neurologists, and also we meet some other families in the online meeting. We need more sleep laboratory in Turkey because a few centers can be performed sleep laboratory. And also this patient have feeding problem, a lot of patients with feeding problem. For this reason, we collaborate the pediatric gastroenterologist and we prepare some videos for the feeding. For example, G-tube replacement, G-tube feeding or NG-tube feeding and replacement. After these videos, our colleagues can give education for the G-tube replacement and feeding. Also, we need some aerodigestive clinic for the swallowing problem. We established aerodigestive clinic for the detect swallowing problem. For example, in our center, these clinics consist of the pediatric gastroenterologist, pediatric pulmonologist, otorhinolaryngologist, swallowing therapist, and the dietitian. In these clinics, we can perform flexible endoscopic swallowing study and also video fluoroscopic study for the detect swallowing problem in these patients. Okay. According to the patient needing, this swallowing therapist arrange patient swallowing therapy. Is the other problem for the neuromuscular disease patient is airway clearance. Airway clearance is very important issue in this patient. We can use manual chest physiotherapy and also mechanical insufflator if patient need it. Some of this patient also need high frequency chest wall oscillation, especially the patient have persistent atelectasis despite the use of the other airway clearance techniques. There are a few doctors experienced for the mechanical insufflator or high frequency chest wall oscillation. For this reason, we organized some meeting or workshop high frequency chest wall oscillation or mechanical insufflator usage. Also, this training, for example, tracheostomy care or airway clearance techniques, we organized some meeting in Turkish Thoracic Society Congress in Turkey. Physical therapy is the main therapy of these patients. Our physical therapist team also organized patient physical therapy according to the patient needing. Also, post-operative care, very important issue in this patient because anesthesia is a very negative effect in this patient. We can perform G-tube replacement endoscopically within in 15 minutes. We try to decrease the anesthesia negative effect. For this reason, we use non-invasive ventilation in the operation room. As soon as finished operation, you can see in this patient. And after scoliosis operation, we can perform non-invasive ventilation effectively, and non-invasive ventilation also decrease the duration of intensive care unit treatment in this patient, and also intubation time. We increase the experience about the non-invasive ventilation and also use this device. We can success extubated or decannulated this patient. You can see some samples of these patients. Our centers is dedicated for rehabilitation patient. For example, neuromuscular disease or ventilator dependent patient. This patient came our centers, other cities of the Turkey, and we gave some training for tracheostomy care, non-invasive ventilation, feeding, G-tube or NG-tube replacement for this patient. Also, some of this patient went to some countries for the gene therapy, Zolgensma therapy. Also, we prepared this patient for the flight or fit to fly. After that, this patient came, go to our cities, but we follow up with telemedicine this patient. For example, according to the blood results or infection, we can arrange this medicine or device settings with telemedicine. This is our Zolgensma treatment results. We have a lot of SMA patient. This is 39 patient, at least longer than 1 year before Zolgensma treatment. This patient took Zolgensma in 22 months. Actually, it is very late. Most of this patient have feeding problem. Half of this patient have chest deformity, and 40,000 of patient have scoliosis. Most of this patient took non-invasive ventilation, and some of use invasive ventilation. Also, common of this patient feeding with NG tube. After the Zolgensma treatment, intensive care unit admission and also intensive care unit hospitalization duration was decreased in this patient. Also, after 1 year of Zolgensma treatment, respiratory support decreased in 4,000 patients. In Turkey, as you can mention, Yasemin, Nursinersen is reimbursement by the Ministry of Health. But Zolgensma is not licensed in Turkey. Risdiplam is licensed, but not implemented in Turkey. Also, we have newborn and screening program in Turkey. What we need in Turkey, a mechanical inexfibulator and a lot of physical therapy device, not reimbursement in Turkey. We need also more doctors with experience with non-invasive ventilation. Also, we need more physiotherapists with experience in neuromuscular disease. But we need especially center specialists in neuromuscular disease. In these centers should be have pediatric pulmonologist, neurologist, gastroenterologist, and swallowing therapist in Turkey. At the beginning of my speech, I said this is a time, this is the team playing game of the management of non-invasive muscular disease. But the most important player of this team actually is family. Family is very important, especially in Turkey, family members playing like a maestro. Thank you for listening to me.
Thank you very much for your complex presentation and for showing us your experience and sharing also your needs. We are talking about complex disease that for sure they need a multidisciplinary approach, and all the speakers emphasize this. The problem is that in many countries this is not still a reality, so we have to fight to have access to integrated care and to multidisciplinary teams in approaching our complex cases. I'm now opening the floor for questions or remarks, comments. Are there any questions? I would like to know, how are you funding your center? You were explaining about your center, but how is it funded?
Our center is private center, unfortunately.
Yes, private center, but how do you find your funds? Yeah, you are the patients paying for their
patients pay for the device or treatment fees.
Mm-hmm. Okay. Thank you. Are there any other questions? If not, I would like to insist again one more time to open your camera to have a group photo because it's very important for the organizers to have this. So thank you for those who opened, and I encourage everybody to open it. There's no danger. Okay. Okay, so we will continue now with the next presentation that I said previously is bringing a question mark. Can artificial intelligence in MRI help us understand neuromuscular and neurodegenerative disorders? And our speaker that will try to answer to this question is Raoul Bouraf. He is a scientist with more than 10 years of research job experience in France. He holds a PhD in neuroscience and neuroimaging. He works in the Center for Neuroimaging Research at the Paris Brain Institute, Sorbonne University in CERN, and Petit Salpêtrière Hospital in France. Raoul, the floor is yours. Thank you.
Thank you very much for the invitation and for the kind introduction. I hope I'm audible clearly, and I hope you see my full screen of the front page of the PowerPoint.
Yes, we see it.
Okay, very good. So I would like to start a bit with my background. I'm associated with the Institut du Cerveau, ICM, which is, just let me see, yes, it moves. And in English, it's called Paris Brain Institute. So in the team, I am in the movement disorders team, MOVIT, and the Center for Neuroimaging. So it's inside the one of the most largest Western European hospitals, La Pitié-Salpêtrière, and also associated with the Sorbonne University in Paris, where I did my doctoral studies in clinical neuroscience. So I would like to divide also my presentation in several folds. So first fold, I would like to show my expertise, like what diseases I work with. And then I would like to show you the tools we use for these diseases to understand the science behind these diseases and how it could be helpful, particularly on the diagnosis side. We are more involved in the diagnosis side. And the potential for these type of methods, these types of tools which are becoming very famous for neurological diseases. What is the potential in the neuromuscular diseases? So let's start with Parkinson's disease, a neurodegenerative disorder, quite a multifaceted disorder, manifests as a motor disorder and worsens over time. And the non-motor disorders are also observed, which is also quite seen, like sleep disorders somebody was talking about as well today. So diagnosis is quite difficult, only possible when the clinical motor signs starts to appear. So from the neuroscience aspect, what I'm interested particularly in is the substantia nigra, which you can see in the animation that it's being highlighted in red. So there is degeneration of substantia nigra, and where there is a dopaminergic neurons, and there is also RN level augmentation in these Parkinson's disease patients. Then another interesting side is the prodromal Parkinsonism, like to those subjects who do not show Parkinson's disease, but they might convert at some stage to Parkinson's disease. So that is related somehow to the non-motor symptoms. That is called isolated REM sleep behavior disorder. So you can see also a sample video from one of our senior heads of department of the Sleep Center of La Pitié-Salpêtrière from the Center of Professor Isabelle Arnaud. So this is IRBD, where it is manifested as a sleep parasomnia featuring unpleasant dreams, such as acting out the dreams in real. And these are characterized by nocturnal violence and increased muscle tone during the rapid eye movement sleep. And it's quite common in synucleinopathy like Parkinson's disease, Lewy body dementia, etc. So these people have a high chance, like majority of them, they convert into Parkinson's disease. And then they start to have these motor signs as well, slowly. So that means there should be mild substantia nigra damage as well, what we see in Parkinson's disease. So at the side of the tool, I would like to show you what is magnetic resonance imaging, which I'm sure most of you are already familiar with. It's from the technology side. It's a non-invasive medical imaging technique that records the changing magnetic field and gives us different kinds of images based on the pulse sequences. That means based on the question, for example, if we are interested to see these neuromelanin in substantia nigra, we'll do a certain kind of image contrast, certain kind of protocol. Then if we are interested in the iron levels in the brain, we'll do a different kind of protocol. And for example, for muscle imaging, for muscle MRI, we'll do a different kind of protocol. So just with this animation, I'm trying to show you how it is. And in this animation, you can see in my right-hand side that how images are captured. So then the mathematical questions can be dealt with, like how many like cubic millimeter, what volume changes, what signal changes, etc. So let's see some examples of these MRI sequences which we deal with. So these are some of the examples of MRI sequences which we deal with depending on the clinical question at hand or research question at hand. So in my case, I'm mostly involved with the neuromelanin type of sequence, which is important for Parkinson's disease and the IRBD, what I showed you, because it's like a proxy marker for dopaminergic neuron changes. Second, I'm more interested is in RN quantitative MRI. That helps us to get the quantification of iron in the brain and perhaps in other muscle area as well. So this makes us think that there is an urgent need to develop these MRI or imaging biomarkers in the field of disease diagnosis in different kinds of diseases. So this could help in monitoring the disease progression primarily, or analyzing the effects of disease-modifying treatments as well, and can help in early stage of the disease as well. So let us now examine such damages at the level of the structure. So like I mentioned about the neuromelanin pigmentation inside the substantia nigra, which can be quantified using a certain kind of MRI technique. So neuromelanin is a dark, insoluble pigment concentrated in the dopaminergic neurons of this structure, substantia nigra. And interestingly, also in the noradrenergic neurons in locus coeruleus. So from the technology side, it is paramagnetic when it's linked with metals, and when you scan the patients, it becomes hyper intense on these kind of protocols, T1 weighted images. So what does it mean? It means that then it starts to highlight that area where you see the substantia nigra. So you can see in these images, it's a healthy volunteer, a normal subject. It shows all the substantia nigra highlighted, and then you have Parkinson's disease patients starting to lose. So any lay person can see visually that there is a loss of substantia nigra, neuromelanin here. So like dopaminergic neurons are starting to lose. So similar for the IRBD as well. So similarly, you could see for the iron changes in the substantia nigra. Like I showed you the sequence QSM. So you pick the QSM's protocol and you are scanning these patients, and then you see that you can quantify iron also in substantia nigra using this QSM technology of MRI. So that could help to compare the patients with the healthy volunteers and see that there is an iron augmentation very easily and non-invasively. Another structure which is implied is very interesting as well, which is locus coeruleus and subcoeruleus. So it's a tiny structure in the bones of the brainstem, and it is also visible using the same technique of neuromelanin. And as you can see here, for this prodromal stage of Parkinsonism, REM sleep behavior disorder, isolated ones, you see that these two tiny spots, one on the left and one on the right, this is locus coeruleus, subcoeruleus, and these tiny ones disappear when they start to have this IRBD. So that is also like a marker for Parkinsonism to understand how these changes happen. So interesting part is to quantify. This is very interesting. When you scan in MRI, after that, you have to use various strong technologies, strong ideas mathematically to segment these regions so that it gives you as precise as possible the results so that we can see the changes. So from the analysis side, if we could see the measurements, how they are done. So when we segment, it gives you something like this. In green, HV is the healthy volunteer. In green, you can see the substantia nigra, same what we discussed. And when the early Parkinson's disease patients are coming to get a scan, you see there is a drop. You can see clearly from my left-hand side, these images, the first row and the second one, you start to see that there is a slight drop in neuromelanin dopamine neurons. And then you see when you start to progress, like this is around 9-11 years of Parkinson's disease. And the second row is around 1 to 2 years Parkinson's disease. So you see that substantia nigra neuromelanin, it starts to disappear. In the lower row, you see without the region of interest. And then afterwards, I'm showing you a TIV segmentation, that is total intracranial volume. You can see the example, how it is done in this MRI. So it helps us to sort of normalize the results so that we don't care about the proportion of the patient. If the patient has a certain head size, or for example, the effect of age, effect of gender, etc. So we divide these values getting from volumes by this total intracranial volume to normalize. So showing an example of locus coeruleus segmentation here as well. So in this is a template where we segmented primarily like an average, and after that you see this is the tiny flash, like left hand side flash of locus, and then you have the right hand side flash of locus coeruleus. So primarily it goes on to find a window. You see in yellow in the right, its right hand side, and red in its left hand side, left side of the subject. And eventually in the window is trying to find the most highlighted pixel. So in the D image, you see red is the left locus coeruleus and yellow is the right locus coeruleus. So this is getting as precise as possible, but it's very difficult, needless to say that. In the second part, you have the iron segmentation here. You see how the nigral iron is segmented in these QSM images. Now let's see about the tools used to segment this, because this is like a very difficult question to find the precision in segmentation. It could be any kind of patient, it could be healthy volunteer, it doesn't matter, but the precision is very important. So that brings us to artificial intelligence. So in artificial intelligence, we particularly use the idea of deep learning. So what is the motivation behind deep learning? It's rather inspired from the human visual cortex, and the biological neuron is the real factor which inspires us to do this. So it's like you need to have little guidance from the programmer. Nobody has to do like proper programming for this, but once the AI is ready, you just send the image and it should just segment right away. That is the basic idea. So one time you program, one time you make it ready, and once it's ready, it has to work on its own and it starts to get better and better. And you can see in this example that how humans perceive a cat in a similar way with like a biological neural network. We try to make the model learn in a similar way with the images, and the model learns. And for example, with the Nigra, it could segment in similar precision as human raters. So what is convolutional neural networks? A convolutional neural network, also called CNN or ConvNet, they are used a lot in the imaging domain for segmentations. So the idea is it's like a feed-forward artificial neural network, and the connectivity patterns, as I mentioned, between the neurons is inspired by the organization of animal visual cortex. So why it's called convolutional neural network? Because it involves various layers inside it, like convolution layer and several other layers. So I would just like to explain you what is convolution layer, why it's called so. So with this example, in this animation, you could see like the brain would learn, and eventually you just send the image in this question, and the model does not know what it is. And just right after learning it using several layers, it could say, okay, whether it's enlarged heart or it's a tumor, it's tuberculosis. And it could right away recognize using this model that, okay, the patient has tuberculosis. That means the model has learned using several images, several MRIs, X-rays, etc., that how to identify these diseases. So let's see how we define these layers. Like, let's see one example of what is convolution. So convolution is the idea is you can see in these layers that is just the idea is to learn by layer using the mathematical convolution operation. So if you have one convolution operation involved, it's called convolutional neural network. So here in this demo, you can see how it scans the image using the filter to conduct the convolution operation. And for example, for tumor segmentation, also it's very similar. You're inputting a tumor, for example, in the MRI, and it goes to several layers, like I mentioned. So first it's called input layer, and you can have one or more hidden layers. It goes to all the hidden layers, and finally in the output layers, it finds you the tumor, what the user is looking for. So it passes through high dimensional MRI input data, multiple layers. Okay, so there is another technique very important in this AI area that is called data augmentation. Why is it being done in the medical domain? Because you cannot have many images for the model to learn. It's not very easy to make it learn with few images. For example, if you are feeding just 50 tumor MRIs, the AI model would have difficulty to really recognize precisely. So then what we do, we get inspiration from other areas of computer vision. That is called data augmentation. So what you see in my right-hand side is these picture of humans. Actually, they are not humans. They are fake photos created from real photos and merged, changed in certain way using all these filters that they start to look like humans. So they are like fake human pictures. So we take inspiration from that, and below that you see one of my MRI images of neuromelanin, and you see that image is quite distorted. So you take a normal image, you make it distorted. It's like a fake data augmentation technique. So it's an analysis technique used to add the amount of data in an artificial way so that the model learns very fast. And it's quite widely used in facial data generation, in face detection algorithms, and becoming very useful, particularly in medical world when we have very less images to train on. So one of the examples I would talk about, which we did for Parkinson's and REM sleep behavior disorder patients, what we published in Movement Disorder recently, we were trying to study this region, which I showed you at baseline, and the drop in 2 years here. So in drop, you can see clearly and follow up that the nigral neuromelanin starts to disappear. And further, we use this segmentation idea to study the spatiotemporal changes as well, where we characterize the topography over time. As you can see here, there was a healthy volunteer, then studying IRBD and early PD, and eventually for the advanced version of progressing PD, you can see that there is a loss of substantial nigra. So eventual question here is the segmentation, that how to segment as precisely as possible to understand not just the whole structure changes, but also territory-wise changes. And that idea is not just applicable in the brain, but also elsewhere. So furthermore, we took the same idea by manually segmenting these images and having a database of manual segmentation and use it in AI model to create by default precise predictions for new subjects. So for example, what I did here, I used 60 images trained and tried to predict it on 176 by preparing an artificial intelligence model. So it's similar idea, you segment by yourself manually, and then eventually it gives you a result, an AI in fraction of seconds. So here you could see an example of how segmentation is done. So here you saw is the precision that in left of the subject is in red, and in the turquoise, it shows you the substantial nigra segmentation precisely done by the AI model. So this method we recently published in a methodological journal, Neuroimage Clinic, and the neurology side for IRBD and Parkinson's patients in movement disorder. So, so far we saw that it's quite clear that AI using MRI biomarker could be powerful for the brain study, for the neurological study. What are the implications? What is potential in neuromuscular diseases? Although there is not much work done in that aspect. So muscle MRI can also provide information that is complementary to clinical, histological, genetic, and all laboratory findings we saw for the diagnosis of neuromuscular diseases.
Mute, mute. Click on this.
I hope you hear me. So muscle MRI allows the identification of various, for example, fatty replacement of muscle tissue, and is quite common in the diagnosis of, for example, inherited myopathies that are distinctive patterns of muscle involvements on MRI. So these patterns are also established by identification of preferential spared muscles, which may not always be clinically observable easily. So in this way, the radiological implications could be very powerful for these patients. So certain patterns, for example, are specific for a particular myopathy, and others are common to multiple disorders. So let's see some examples, some demos for the implications in neuromuscular diseases. So this is a study recently published in one MRI methodological journal, where they're trying to use AI to segment the thigh and leg muscles, done by a collaboration between Italian and Swiss team. So they are using a protocol very similar to what I showed you in QSM, that is quantitative MRI protocol. It's called GRE sequence. So what they did, they took 6 healthy volunteers and 30, altogether 48 patients with neuromuscular diseases, and the patients were scanned longitudinally 3 times. The scan time is quite tricky, it is quite long, but in brain MRIs, it's quite lesser comparatively, like it's 5-6 minutes for neuromelanin, which I showed you. But here it took them 20 minutes for the thighs and 15 minutes for the leg. So altogether, they had a huge image data set, as you can see, like the slices of images done manually by the experts. So the idea would be to use all these 770 thighs, 330 leg slices as input for the AI. So let's see an example here. In the upper side of the A image, you see the thigh, and B, you see the legs, and you have all other labels. So this is a proper illustrative example of the MRI scans done there, and the corresponding muscle manual segmentations. So using this, they would start to predict. Let's see how is their prediction result. So you see in my left-hand side, the thigh, and my right-hand side, the leg. So in the training data set, what they use, it was very highly accurate for them. So you see, this is human side in my left, and DNN is deep neural network, DNN. So you can see in the segmentation that DNN could replicate very similar. So DICE is a measure for similarity index, like done by the humans and done by the robots. So what is the similarity? So DICE, you could see for training is 96.31%, which is quite remarkable, I would say. You see similar for leg, it's above 90. So for case one, you see, visually, if we see, it's quite similar. Similar you see for case two, but these are for training data set, the data set they used to train the AI model. Then you see for validation, the ones which were not touched during the software preparation, like during the AI training. So these are images coming from outside, and they are just feeding it in the AI to have the results. So here also you see it slightly goes down, but it stays more than 90%. And you see case one and case two and the case three as well, not quite bad, all quite high, more than 90%. So I would say quite exceptional result with the overlap between manual and the artificial intelligence segmentations, showing the power of AI in MRI for the neuromuscular patients as well. There is another study done in Australia. It is by using the T1 weighted MRI, is like one of the classical conventional MRI system, like using cerebral palsy patients, involving 20 children where 6 of them had cerebral palsy. And as it is quite known that the muscles of children with cerebral palsy are smaller than muscles of typically developing children. So that could be a good marker for MRI to compare how it works out. So even this AI model was quite powerful, and they could replicate, as you see, DICE as a similarity index between manual and automatic segmentation of 90. For the typically developing children MRI, and for cerebral palsy of 0.85. So it's still excellent, I would say. So in this example, you could see A is a typically developing child, and B is the cerebral palsy, and there is C and D are the corresponding manual segmentation, respectively, in axial slice, approximately midway between the ankle and the knee. And you can see the precision is quite strong. Like if you see from an expert eye, for as radiologists, is quite remarkable how the segmentations are done. So as it is well known that they differ in shape, position, size, volumes, this could be a very, very strong marker for the future of neuromuscular disease patients as well. So these examples are also very interesting. These are what is produced by the AI models, example segmentations of muscles and bones from MRI scans of the 4 participants, as you can see, 1, 2, 3, and 4 in rows. And these are produced by 6 AI models, which these people used, and along with the human raters. So first, this is the input MRI, the grayscale one, and this second column is the manual segmentation done by the expert, apparently by radiologists, and the others are replicated by AI. So with human eye, we could feel that the replications are quite strong by the AI. And however, this example shows how first and third rows the scans were kind of not so well segmented, and the second and fourth row examples are quite a well segmented one. And then I have one 3D rendering images for these. Also, the 3D unit is AI example. Second is also done from AI. Third is also done from AI, and fourth is by manual segmentation. So with these 3D renderings of segmentations of MRI scans, with the AI predictions compared to human raters, you could say, it produces very accurate segmentations, actually. And all these 3D renderings were taken from same viewpoints, indeed. So you saw the potential of artificial intelligence, how it in the domain of neuroimaging, as we do, it can also be used in muscle imaging in the similar way. And it's an area not so tapped into these days by the medical researchers. Final words. So we saw multimodal different types of MRI biomarkers, and particularly coupled with AI segmentations. So we saw it's promising and potentially transferable to different skeletal muscle groups as well. It is direct, and being artificial intelligence is non-rater dependent. Once you have your AI ready, there is no rater implications in that. It's non-invasive because it's MRI. The patients are not quote unquote touched in these cases. So these automated methods can also help monitor muscle growth and evaluate the effectiveness of interventions, and possibly the first step towards pre-symptomatic diagnosis of these diseases one day in clinical practice. Thank you very much for your kind invitation. I really appreciate it. And thank you, EAMDA, again for everything. And just a note of thanks for my friends and colleagues. And I'm very much reachable if you would like to collaborate, if you would like to discuss, and if you have questions, even afterwards. I have my website, my Twitter, and my email also here. Thank you very much.
Thank you very much for your presentation. And we open the floor for questions.
Can I make a couple of
Yes,
comments? Question. Raul, thank you very much. This is very invaluable. Actually, your presentation is invaluable. I'm a mechanical engineer and a patient advocate since my daughter has a genetic related disorder. So over the past 2 years, I have been, trying to understand what's going on this field. And as an engineer, I have understood that AI is one of the biggest things that have been helping related to gene therapies and all these other stuff related to the genetics. And also I have come across a British-based company, DeepMind. One of the executives is from my university in Turkey, and I have managed to reach him. So actually, this is super important during our journey to be able to reach the drugs about all these genetic related diseases. AI is super important. So thanks again, and I'm glad to see such a presentation and an expert from this field, because I think all patient advocates also have to find and seek solutions to support experts like you on this field, because I'm pretty sure that this will have a big impact on finding the cures, to focusing on AI in the future. Thanks again. Also
Thank you very much.
To find.
Thank you. I appreciate. Thank you.
Thank you very much. Any other comments or questions? Don't be shy. Okay, so thank you very much again, and we'll continue the presentations. And our next speaker is Karolina Podalska. She is going to speak about the care coordination and transition of care for neuromuscular disease patients. We all know that care coordination and effective transition of care are essential for a high quality of care in rare diseases and neuromuscular diseases, and we are looking forward for your presentation, coordination of transition of care for neuromuscular patients. Karolina, the floor is yours.
Thank you very much. Hello, everyone. I hope you can hear me. I will just ask Arabella to share the presentation, please. So maybe before you share the presentation, I'll just briefly start. My topic is, as you mentioned, coordination of transition of care. My name is Karolina. I am a clinician from the Czech Republic and also the head of operations in Parent Project Czech Republic, which is a patient organization that is focusing on helping patients with neuromuscular diseases, especially Duchenne muscular dystrophy in the Czech Republic. Why I chose this topic. I think transition of care is extremely important. We made a questionnaire a couple months ago asking patients about what words come to their mind when you say transition of care. And unfortunately, the results were very alarming. We heard fear, uncertainty, afraid, desperate, and so on. So we do know that it's a big concern in our patients' minds. And the project that we started is called Coordination of Transition of Care, because what we found out is that what they really need is just someone who will ask them good questions about what they need and who will focus on building a network of specialists who can take care of them.
Carolina, so sorry. I think that I don't find your presentation. Did you send it by email?
Yeah, I sent it twice, but okay, I'll just try to
Maybe
share
I
my
didn't
screen.
get it. Or if you can send it right away, I will just open from my laptop.
Okay, I'll do that.
Yeah, sorry about that, but I know I've searched it already earlier today just to see if you sent a presentation, but maybe I didn't receive the email. So
I send it to you now. Okay. The situation in the Czech Republic is such that we have absolutely excellent pediatric care of neuromuscular patients. It is really world top. But when the patients turn 18 or 19, what they get is just one name, one email, one person they can turn to, one neurologist who really doesn't have time to take complex care of the patient, who asks them about new problems that they have, asks them about the need of a wheelchair, gives them corticosteroids if they need it, and that's it. And the neurologists really don't have the capacity, the time to take care of those patients in a complex way. And really, ever since we have so good care in pediatric system for our patients, we have a population growing of need, a population of adult patients that we didn't have before, especially of Duchenne muscular dystrophy. So, and you might all know that really this disease is extremely complex, and especially when you get older, the problems that come up with your heart, with your lungs, the needs for sleep studies and for ventilation and for cardiac medication and like a GIT tube, gastrostomy. Those are very specific needs that not everyone really understands, not every doctor. And the experience with Duchenne patients is very low in the Czech Republic, especially. We're a small country. So what we've came up with, yes?
Sorry, I still didn't receive it. So maybe it is a problem still with my email or anything, because it didn't came. Okay. Can you very, very quickly forward it to the email you see in the chat? It's our IT team there. Okay. And they can do it.
Okay.
So
I will do that.
Yeah. So this one, yeah. Or if you think you want to try to share your screen again, but I don't know if the connection will be then. It's interesting because I've received your emails, but not a presentation, so I don't know why.
I don't know either. Okay. I send it to the email. So in my presentation, I briefly speak also about our organization. What we offer to our patients overall are stays, week stays, weekend long stays, where they can meet other families and also a team of experts. We also offer mobile care. We come to our patients to their homes and discuss with them their needs. And we have a lot of telemedicine projects working online and over the phone. And so the role of coordinator of care is really what it looks like is that the pediatric center contacts me as the coordinator when the patient is turning 18, let's say, and they tell me, okay, the patient will come in and we will examine him for the last time, and then they will have to transition. So they'll be yours. And I come and meet the family and discuss with them what their life looked like until now, and what was like a big issue that they needed. Thank you for sharing it. So we can move maybe 2 slides or three slides. Yeah, and next. Yeah, thank you. Thank you. Yeah, that's a good slide for me. So what I do is I contact the family, I talk to them, and they told me that, for example, the patient has a lot of problems with their lungs, they need ventilation, or another patient is more needing of cardiac care, or someone else has problems with nutrition. So I really try to do individualized care and really just focus on the area that is most needed for them. And then we come up with what suits them in terms of appointments. I ask them if they are able to come once a year to a hospital to have an appointment, if they all need the appointments in one day, or if it's okay for them to have it in 2 or 3 days. And then we work with that. And what works really very well is that they know that I'm one clinician that knows them, knows their story, and they can call me if they need to, and I will know what is behind their story, and I'll know more. Of course, really none of the other doctors have the time capacity to take care of them in this way. And I think it helps the doctors as well to have one coordinator, because the coordinator can also discuss in acute situations. And when the family calls me and tells me we have this kind of problem, a fever and arrhythmia or something, we can talk it over within 2 or 3 minutes, and then come up with if they need to go to the hospital, if they need to go within 10 or 20 minutes, or if it will wait for a few hours, or if they should just wait to the next day, and what they can do. So we do kind of a triage situation and discuss with them. And they feel much calmer because they can talk to someone who knows them and who knows neuromuscular patients. So I think it's a system that works for the families especially. But as I write here, I also try to build a network of specialists who will take on those patients, and it's a good position to be coming from a patient organization, not to be a parent, but to be someone officially from a patient organization, and come asking for this kind of solution and asking for cooperation. And rarely anyone says no, actually. So that's really good news that we have seen just a lot of positive reactions. And if I give the doctor some numbers of how many patients they will take on or so, then they usually calm down, because if I ask them to take care of Duchenne patients or neuromuscular patients, they imagine a few hundred of patients, which is definitely not possible for them. But if I tell them it's usually 10 or 20 patients per year, then it seems doable much more. Next slide, please. So this is a picture from our stays. It actually helps a lot to meet the families in person at those stays, or of course when they come to the hospital at their hospital appointments. And what I also mentioned here, that not only in this project do we take care of patients who are transitioning, but also of patients who are already adults but do not have complex care. So if they sometimes have very to me, simple issues. For example, or it seems simple, they don't have an eye doctor. They don't really need a special eye doctor for neuromuscular disease, but they don't need an eye doctor where they would have accessible examine rooms. So this is something that I really can solve much easily than anyone else. I can ask other families where they go, and then call the office and just ask them if this patient can come. So the families are overwhelmed with so many information and so much to do. And just one person of a coordinator can help a lot, making those phone calls and helping them to organize the appointments. What is also great is that I have much more time to discuss with patients the treatment, why they can or cannot join a clinical trial, what it means, what they can expect from research, what they can really expect from the future of these diseases, and what they should prepare for. So we have much more times for discussion, and that's, I think, also crucial for the family. Next slide, please. I also put here some numbers. So in Czech Republic, we have about 300 patients with Duchenne muscular dystrophy, about 400 of children's neuromuscular diseases. We, of course, have other organizations taking care of neuromuscular patients. So, but if the patient really contacts us, what we provide them is specialized care, individualized care, if it's in our capacity, and it usually is. We have about 150 members of Parent Project Czech Republic, and about 20 families are part of the coordinator's care, but it varies also. Sometimes someone reaches out only once or twice, so I don't really count them as part of this project, but it's from my point of view, it's just good to know that you have someone to call in case of trouble or need. We have been doing this project for 3 years. We started with only 5 families, with 5 people who are transitioning that year. And we have about 10 specialists that joined our adult care team since. So I think that the network is really building up, and that it just helps the patients a lot. What is also interesting is that sometimes the coordinator's care is needed only in the beginning. So you just set up a system in the beginning, and the family knows that someone discussed it with them, that the care is set up, that they have a cardiologist, that they have a pneumologist, endocrinologist, and so on. And then they are already fine calling there, making appointments themselves. So they don't really need the care throughout the whole life, but in the beginning, very much so. Next slide, please. So this is just an overall information on how to start. I really want to encourage you, if you feel that there's a need in your community that is unmet, try to come up with innovative ways to solve it. It took us a long time to find out what works the best with our families, but I think that we're on a good path now, and it was all worth it. And to us, it is very frustrating that we cannot change the adult care system in a few months, let's say, and that we cannot build an adult complex care in the Czech Republic straight away. But this is a solution that is temporary, and that is helping the families now, which we're very grateful for. Of course, it's not easy to try to find financing for your project, but also there are resources to look into, and a lot of, for example, pharmaceutical companies are willing to just help you start with an idea that you have. And of course, our ultimate goal, as I mentioned, is building an adult complex care center. So that's what we're working on in the long term. But the coordination of transitional care is really right at the moment solution that we have. I think it's the end of the presentation, and I'm just glad I could share this project with you. And if you have any questions or want to ask about experience or about what we did and how to implement it in your country, then you can write to me or tell me your question now. Thank you.
Thank you very much, Carolina, for sharing this. And yes, we are open for your questions or comments.
One comment and one question. One comment, it's great. I really love it. I hope you'll be able to have more and more coordinators accompanying more and more families, because I think that's really a very important way to make sure that patients are able to have all the support they need and all this coordination, as you said, because that's really needed. Just a question, in the accompaniment or coordination, do you implicate psychologists, and is the access to psychologists easy or difficult in your country?
That's a wonderful question. We have 2 psychologists as a part of the team, 1 psychologist and 1 psychotherapist. What is very interesting for me is that when people join Parent Project Czech Republic, they all say that they need psychological support. They all mention it really in the first phone call. Everyone does. And then when I give them the number, they never call. Like really, like one in 10, not really 1 in 10. Just that they just don't call. They don't reach out. So I think that when it's not like handed to them and like push on them, they don't accept it well, because they feel that they don't need it, even though they probably don't know what they're missing. We do psychological work with our families at the stays that we organize. We do them with the parents, with siblings, with patients. And that works really well for us, because it's in a form of discussion or even play for the kids. But whenever you call it psychological support or a consultation with a psychologist, no one really reaches out. So it's hard for us. But we actually hired one new person in our team who has experience with palliative care. And she's not a psychologist, so she is a little free in the rules of how to make connections with patients, and it works much better. So if she doesn't introduce herself as psychologist, and if she's part of the hospital already, so if she meets with the family in any other way, then it opens the door for next cooperation with her. So yeah.
Okay, and as a quick other question, in France, we don't have a similar system, but we also have professional accompanying the family in all their need to also for inclusion in school, access to university, access to employment. And what we realize is that it's very hard to sustain and to maintain financially when it's just on a small number of patients. So in France, we do it for all neuromuscular diseases, and we try to convince the government to open it to all situations where people have some kind of dependency. So even elder people, Alzheimer, etc. How do you see it? Do you plan to open it to more pathologies, more all neuromuscular diseases, maybe other kind of rare disease?
That's a good question also. As we get better in our work, of course, more patients come to ask for help. What we really work on in the long term is advocating for case manager kind of situation in the Czech Republic, which is not available now. But we really have right now a questionnaire, a research going on to find out just how much it would be really needed to have case managers and like individualized care. So that's what we're aiming for in the long term. But in terms of financing, and it's very hard to just look into like big grants, and we have a lot of patients coming in, of course. But as of now, we can handle it. And also what is helping is the community sharing. So we just started a community that will be on the website, like a discussion, open discussion kind of thing. And when the parents share their experience, it's also so valuable, or the patients, of course. So if it's a little like organized, and if it's not like, where do you go to the movies kind of questions, then I think it works well as like inspiration to other families. And sometimes questions get solved there without the experts needed.
Thank you very much. Just I've got a contact with SMA Czechia. So if maybe there could be some kind of collaboration with the SMA population, I think this could be done.
Yeah. Okay. Sounds good.
Other questions? Comments? If not, I will try to summarize what we discussed today. It's very interesting how similar we are and how different healthcare systems we have. Even if we have many similarities in Eastern Europe and many challenges, we share many challenges. It was a very interesting and very inspirational day for all of us, I think. And we started in the beginning and found it about the many projects that EAMDA is implementing to improve the quality of life for patients through summer camps, rehabilitation programs, and social activities. We also found about innovative projects like the radio program, the think tank meetings online on different topics, and about the nice project on NFT art gallery. Then we had a presentation on Safe Polymed project, which is a research project with the goal to increase drug treatment safety, to provide physicians and pharmacists with innovative tools to define, assess, and manage drug interactions. We found more about the International Clinical Trials Registry platform for DMD, and also about the advances in clinical trials in non-Duchenne muscular dystrophies. Our next speaker has spoken about SMA, from a fatal to a treatable disease, as the introduction of new therapies for spinal muscular atrophy has changed the clinical landscape, transforming SMA from a little to a treatable disease, and found out about the stepwise approach for newborn screening for SMA patients, babies, newborns in Serbia. It was a very inspirational presentation just before the lunch. After the lunch, we found out how the interdisciplinary approach, integrated and palliative care, improved the quality of life for patients with ALS in the last 20 years in Ljubljana. The next topic was genetic testing, as understanding testing methods, the link between genetics and neuromuscular diseases, interpreting complex results, and dealing with the ethical, social, and personal issues that arise for patients and families is critical for their care. And that genetic tests are helping not only the patient, but also the family. Then we found out that caring for patients with neuromuscular disease is challenging, and respiratory care is of the utmost importance because of its major determinant for quality of life and survival. We had great presentations on the management of respiratory symptomatology in patients with neuromuscular disease from Turkey, and also some presentation that show us the reality and the projects that are implemented in their center. Next presentation brought up an important question, like can artificial intelligence in MRI help us understand neuromuscular and neurodegenerative disorders? And new studies demonstrate the advances being made in diagnostic and quantitative muscle imaging, including the incorporation of artificial intelligence for image analysis. And the last presentation proved one more time, if was needed, but we all knew how important is the coordination of transition of care for neuromuscular patients for high quality of care. Bridging gaps between medical, social, and educational services, along with transition from child to adult care, training and education may lead to more effective care coordination and transitions in the future. All day we heard about the importance of multidisciplinary approach and collaboration, and we really have a lot to learn from each other, and I think it was a great conference today. So thank you all for your participation, and I give the floor to Arabella to inform you about next steps today.